beautiful campus of Mahe, Bengaluru, as we plan to celebrate the occasion of the Postgraduate Orientation Day program of MIRM, ARAMBH. I welcome the batch 2024 new students today, as you are here to discover your potential, to evolve and shine. This is Dr. Uttara Chakrabarti, and I will steer you through the entire program along the day, along with my colleague, Dr. Meenal Francis. Without further ado, let's start the day with an invocation to the Almighty. May I call upon stage Dr. Raghavendra Prabhu, Professor Shashidhara, and Professor Jyoti Prasanna to first light the lamp. Can two students come up on stage to light the lamp along with our dignitaries today? Any one of you, come. Okay. One master student and a PhD student. I can see some of you at the back. Please come, any one of you. Thank you. You may please take your seat now. We all await to start the day with a Saraswati Vandana, a performance given by our own students, Shritama and Purvi, who will grace the beginning of ARAMBH on this day. Hello, hello, hello.
जय जय देवी जय जग जननी जय जय सरस्वती माई जय जय देवी जय जग जननी जय जय सरस्वती माई जय 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 भवानी जय शर्वाणी जय जय भवानी जय शर्वाणी जय त्रिभुवन सुखदाई जय जय भवानी जय शर्वाणी जय त्रिभुवन सुखदाई तेरी मायानंत पार जाको को नहीं पाई जय जय देवी जय जग जननी जय जय सरस्वती हस्तकमल मोबीन बजावे हस्तकमल मोबीन बजावे या में सब सुर गाई दूजे हाथ विराजत पुस्तक वेद श्रुति उपजाई जय जय देवी जय जग जननी जय जय सरस्वती माई जय जय देवी जय जग जननी जय जय सरस्वती तेरी रूप भयो सब विद्या तेरी रूप भयो सब विद्या सुर नर चरण नमाई पद्मनाभ प्रभु चरण सरोरुह सेवक के मन भाई जय जय देवी जय जग जननी जय जय सरस्वती जय जय सरस्वती जय जय सरस्वती थैंक यू
Thank you, Shritama and Purvi, for such a wonderful rendition with the Saraswati Vandana. May I now call upon the three dignitaries on stage, Dr. Raghavendra Prabhu, Professor Shashidhara, and Professor Jyoti Prasanna. Please come up on stage. I shall now call upon Dr. Raghavendra Prabhu to deliver the presidential address to all today in this occasion.
So good morning, everyone. The chief guest of today's function, Professor Shashidhara, the Dean of MIRM, Dr. Jyoti Prasanna, esteemed faculty members of MIRM, staff, students, and the parents of the first year PG students of MIRM. A hearty welcome to all of you to this wonderful campus of Mahe, Bengaluru. So it is my pleasure and honor to be here for the orientation program, ARAMBH, of first year PG students of MIRM. So along with the presidential address, I'll just take you through a glimpse of Mahe as well as Mahe Bengaluru. So regenerative medicine is at the forefront of scientific innovation, holding the promise to revolutionize healthcare by repairing, replacing, and regenerating damaged tissues and organs. Your decision to pursue advanced studies in this field reflects a commitment to pursuing the boundaries of medical science and improving the quality of life for countless individuals. At Mahe, we provide you with a rigorous and supportive environment that will enable you to excel in your studies and research. Our distinguished faculty members are leaders in their fields, committed to mentoring and guiding you through the complexities of regenerative medicine. Engage with them, seek their expertise, and take advantage of the cutting-edge research and resources that is available to you. As you embark on this journey, I encourage you to embrace the following principles: scientific curiosity, rigorous research, ethical responsibility, collaboration and interdisciplinary research, and global perspective. So during your time here, I urge you to immerse yourself in rich academic and research opportunities available to you, participate in seminars, engage in collaborative projects, and attend conferences, and network with professionals in this field. So these experiences will be invaluable as you progress in your career. Your work in regenerative medicine has the potential to transform healthcare and offer hope to patients facing previously untreatable conditions, whether you focus on them or stem cell research, or tissue engineering, gene therapy, or any other area within this dynamic field. Your contributions will be a cutting edge of medical science. As I already mentioned, I just want to take you through Mahe and Mahe Bengaluru campus. So this is a glimpse of Mahe. We have around 4 campuses, Manipal, Mangaluru, Bengaluru and Jamshedpur, and 2 offshore campuses, one in Malaysia and one in Dubai. We are offering 370 plus programs in over 36 plus disciplines with the help of around 25 institutions and 15 teaching departments, university teaching departments, along with around 130 research centers. The architect of Manipal, Dr. T.M.A. Pai, he had a vision of starting this Manipal education. So in 1953, he started with the first medical college of this country, the self-financing medical college of this country, which is Kasturba Medical College, Manipal. So from then, many colleges, many institutions, many teaching departments got added, and at present, we have almost 45 institutions and departments offering various programs in various disciplines in our university. His son, Dr. Ramdas Pai, the present chancellor of Mahe, he took Manipal or Mahe from Manipal to other places of India as well as to abroad. So he made Mahe as an international university town, or Manipal as an international university town. In recognition of his achievements, both Government of India and Government of Malaysia, they offered him the prestigious Padma Vibhushan Award by the Government of India and Datuk Award from Government of Malaysia. So these are our campuses. So the main campus is in Manipal. Then we started a campus in Mangaluru, then Bangalore, Jamshedpur. So in Jamshedpur we started a first the private medical college in partnership with Tata Steel Limited in 2021. And the Bengaluru campus we started in the year 2020 with Srishti Manipal Institute of Art, Design and Technology. At that time, so initially this Srishti, it was offering programs from almost over 20 years. Then Mahe took over that institution. Earlier it was under various other university. It came under the umbrella of Mahe in 2020. Then we have offshore campuses in Dubai and Malaysia. As I already mentioned, we started our journey in 1953. We started the first self-financing medical college of the country, which is Kasturba Medical College. And from then we added the institution and teaching departments almost every year. And in 1993, we got a status of deemed to be university from UGC. And in the year 2020, so the Ministry of Education, Government of India, so offered the status of Institution of Eminence for Mahe along with other 5 institutions in the country. So again, just a glimpse of Mahe. We have almost more than 10 top-ranked institutions in the country with over 3,500 plus faculty members, with over 250,000 plus distinguished alumni. So other details you can see, we have around almost 20,000 plus hostel beds across all our campuses in India. Just a update about the rankings. In the national scenario, NIRF has ranked Mahe as 6th university in India, and In THE Impact Ranking 2024, Mahe has been ranked 15th worldwide in gender equality. And when it comes to the UI Green Metric Award, World University Ranking 2023, Mahe has been ranked number 1 in India and number 39 in global suburban category. So these are some of the certifications received by various institutions of Mahe, be it NAC. So NAC has accredited Mahe with the highest grade of A++ with a CGPA score of 3.65 for 5 years, which is valid till May 2027. And there are other accreditation bodies. They gave certification and accreditations for our various programs across our university. Coming to Mahe Bengaluru campus, the groundbreaking ceremony that has happened in the year 2020, October 2020, we started with this academic block, the first academic block. It is called as academic block 4. So we completed the construction of this academic block 4 in the year 2022, and we started operations in May 2022 with Manipal Institute of Technology, Bangalore. And currently we have 5 professional higher educational institutions and 3 university teaching departments offering various programs in various disciplines in this particular campus. With the completion of this year's admissions, we have almost 7,100 plus students studying in this campus. So 400 plus faculty members are serving these institutions. We have almost 3,800 plus hostel beds, and the students are staying within the campus. And when it comes to the data, around 20 to 30% of our students are day scholars. So all others are staying within the campus. So just for ease of operations, Mahe has restructured all the organization into 3 parts. One is Faculty of Health Sciences, the second one is Faculty of Technology and Science, and the third one Faculty of Management, Humanities, Liberal Arts and Law. So in Mahe Bengaluru campus under Faculty of Health Sciences, we have 2 institutions. One is MIRM, this is Manipal Institute of Regenerative Medicine, started in the year 2007, and Manipal College of Health Professions, offering allied health programs, which started in the year 2010. When it comes to Faculty of Technology and Science, we have again 2 institutions. Manipal Institute of Technology, Bengaluru, started in the year 2021, and Srishti Manipal Institute of Art, Design and Technology, started in the year 2020. So earlier, this Srishti Manipal Institute, which we had around 6 campuses in Yelahanka. So but now, last year, so all those 6 separate buildings, they were operating in 6 different buildings. So all we have shifted to this particular campus, and all 8 institutions are currently operating at Mahe Bengaluru campus. When it comes to Faculty of Management, Liberal Arts, Humanities and Social Sciences and Law, we have 5 institutions and teaching departments, all started in the year 2022. So TFI Management Institution Bengaluru, which offers BBA and MBA programs. So Department of Liberal Arts, Humanities and Social Sciences offers programs in psychology, English. Then we have journalism, media communication. So Department of Public Policy offers programs in MA in Public Policy. Department of Commerce, Bengaluru, which offers again BCom and MCom programs. And Manipal Law School, which offers BA LLB, BBA LLB and LLM programs. As I already mentioned, we are planning to have 6 or the 8 academic blocks in this particular campus. So 4 academic blocks will be, we are operational now, and 4 more under construction. So academic block 2, which is for the Manipal Law School, DLHS and MIRM, so that is already operational. Academic block 3, which is for the SMI, Srishti Manipal Institute. Academic Block 4 and Academic Block 5 is for Engineering College, which is the Manipal Institute of Technology, Bengaluru, and Academic Block 6 for the TAPME and Department of Commerce. So with the completion of all 8 academic blocks, it can house 20,000 students in campus. So we are hopeful that all these 8 academic blocks are completed by 2026. When it comes to hostel block, again we are planning to have 4 hostel blocks. 3 are already operational. The 4th hostel block, it will be operational from August 15th, 2024. With the completion of all 4 hostel blocks, we'll be having around 5,622 beds in the campus. So these are some of the central facilities: the food court with the auditorium, the indoor sports complex. So both these facilities, the food court and auditorium, that is already operational. Indoor sports complex, it will be operational from August 12, 2024. Administration block, again it is under construction. It will be operational from by the end of this year. The multi-level car parking, again it is under construction. Once it is completed, it can house around 1,700 cars in this particular building, and other service block and utility blocks already in operational. So these are again some of the central facilities: the AB4 auditorium, the one which you are sitting here, the library. Again we have library at multiple locations as of now. Again we are having to have the central library at one location. Again that is under construction. By the end of next year, the central library will also be ready. Dr. Ramdas Senpai Convention Hall, which can house around 1,100 students at a time. The state-of-the-art auditorium, again it is operational. The main food court, the marina food court, amphitheater, so all these are currently operational. So some of the outdoor sports facilities, we have a cricket ground, football ground, 2 basketball courts, tennis courts, 2 volleyball courts, 4 cricket practice pitches. Along with this, we also coming up with a marina. As I already mentioned, it will be operational from August 12th, 2024, within 2 weeks' time. So it has various facilities: basketball court, volleyball court, table tennis courts, your the badminton courts, steam and sauna bath, and all gym facilities, again in multiple levels, and rooftop tennis courts. So we are happy to share the information that around 24 researchers of Mahe, they are in the top 2 percentile of the researchers in the world, and from that around 5 researchers are continuously in this list from the past 3 years. So some of our distinguished alumni, we are proud of our alumni with their achievements. We are also hoping to see you all in some time in this particular list. So Dr. Ranjan Pai, the president of Mahe Trust and the chairman of MEMG, he is at the forefront. So he is leading Mahe and other Manipal group institution. So he is supporting, he is giving his, the Bengaluru campus is his brainchild, and he is both Dr. Ranjan Pai and Dr. Shruti Pai, they are continuously monitoring the progress of this particular campus. So with this, I will end my speech. So once again, welcome you all to Mahe Bengaluru campus. Wish you all have a fruitful stay for the next 2 years for your PG studies here at Mahe Bengaluru. You can also continue your studies here at MIRM by pursuing your PhD. We are also offering scholarships. We are having around more than 500 scholarships offering across all the institutions in the university. So hope to see you all in future as well. Wish you all the best. Thank you very much.
Thank you, Dr. Prabhu, for an in-depth information about this new campus, which is something very important for all of us to know about. May I request now Dr. Jyoti to please, as a token of appreciation, hand a sapling to Dr. Prabhu. Our next speaker, may I request and welcome Professor Jyoti Prasanna, Dean of MIRM, to address this gathering and officially announce the release of the MIRM newsletter, Rejuven, on this occasion. Your stage.
Thanks, Dr. Uttara. Good morning, everybody. Welcome to the Mahe Manipal family. I call it a family because our students, our faculties are there with us for lifetime. I can see a lot of nervous enthusiasm, lots of questions, happiness, all mixed together on the faces of parents and students. But I, as a Dean of MIRM, I'm here to assure you that we are going to take you through this journey, mentor you, and make sure that you emerge as confident individuals. But at the same time, it's also your responsibility to uphold the scientific and academic integrity of this institute, which Mahe and MIRM is known for. I also take this opportunity to thank Professor Raghavendra Prabhu, who has always been very, very encouraging, especially when it comes to MIRM activities, and his one registrar or deputy registrar who is always available on phone 24/7. And we were on the midst of a shift in the month of March, and right from the smallest problem, be it AC duct, water leakage, to everything, he makes sure that people are available to assist us and help us. And thanks for your presidential address, Professor Prabhu. And he's a truly, truly humble administrator who is always available, especially for MIRM. And he's a good cheerleader for both small and bigger accomplishment of MIRM. I would also like to thank Professor Shashidhara, the director of NCBS, TIFR-NCBS, to take out his time to come on the Orientation Day program. Professor Shashidhara's journey had been very interesting, actually, apart from the fact that he's a director of NCBS and he's a professor at IISER Pune, and he also has an honorary professor position at Ashoka University. He started his journey actually in the field of agriculture and plant genetics. And then he went into evolutionary biology and then Drosophila as model systems to ask questions about cell size, spatio-temporal decisions when you're talking about body organization. And now he's integrating these evolutionary principles to understanding cancer. And that's literally very, very inspiring, because when you hear, as we all sit in different programs, we have the stem cell biology program, we have the translational neuroscience program, and this translational immunology program. And like during our interview sessions, we had questions like whether this is good, that is good, what should I choose? But on this stage, I want to tell, especially Professor Shashidhara's journey says that science is becoming truly interdisciplinary. And the scientific quest, or the scientific, your inspiration, taking from different model organisms to ask broader questions with respect to disease is the key. And I'm sure that irrespective of whichever program you have joined in, by the end of it, you would be able to find questions, interesting questions and answers on different aspects of disease. So Professor Shashidhara has a lot of feathers in his cap, apart from the fact that he's an able administrator. He's an SSP Prize awardee, an elected fellow of 3 Science Academics of India. He's also an elected member of the European EMBO organization. Professor Shashidhara has also served in the past as Vice President of Indian National Science Academy, and Shashi has also been involved in science education and in policy decisions in science and technology. It's also very interesting that he has conceived and initiated the co-project of IUBS, which is an international project on climate change education. I think looking at what all we are facing with respect to climate crisis, I think this is a very, very important aspect which he is contributing to. Thanks, Professor Shashi. Also, on this occasion, we would take this platform to launch a second issue of our newsletter, Rejuven. Rejuven would not have been possible if not for the untiring efforts of our editorial team, Professor Anujit Kumar, Professor Uttara, our assistant editors, which constitutes of our PhD students and master students, and our designers who have really put together the whole thing, right? So the newsletter is not a platform to brag about whatever we have accomplished. It's also a platform where we would engage with our stakeholders. We would want to send it across that we are a highly interdisciplinary collaborating institute. We are open to criticisms, we are open to suggestions, and I would want all our students, faculty, and parents to go through this newsletter and give their valuable suggestions for that. Thank you. With this, I would request the dignitaries to launch our newsletter.
So may I call the editors of the newsletter team and Professor Anujit to come up on stage. I'm excited to announce that our students have really come forward this time to contribute to this newsletter in terms of articles and quiz crosswords that they have written and prepared for the newsletter, and as a result of which we decided that MIRM will provide them with a token of appreciation, certificates and cash awards, which will be given to them as I call the names. Can I call upon stage Deepak? Is Deepak here? The next recipient of this would be Kaustubh. Is Kaustubh here? Please come up on stage. Professor Shashidhara can hand over the Next would be Alan Brito. The other recipients are Rishani, Trisha, Samiksha, and Varsha. I don't think the students are here to receive the certificates today from you. So with this, we can finish the certificate giving session to our students. And we would now request our dignitaries to please take your seats. As you all heard of our guest of honor, from our Dean, Dr. Jyoti Prasanna, I will take another 2 to 3 minutes to formally introduce the speaker before we invite him on stage to give his lecture. Professor L.S. Shashidhara, a leading scientist in genetics, molecular biology, and evolutionary biology, has significantly contributed to cancer research and scientific education in India. As a founding academic member of IISER Pune, Ashoka University, he has played a significant role in their growth. He did his undergraduate and postgraduate studies in genetics, plant breeding at the University of Agricultural Sciences, Dharwad, India, and he pursued his PhD at the University of Cambridge in UK. He continued his academic pursuits at the University of Cambridge as a postdoctoral researcher, where he began his seminal work on the function of Hox proteins in appendage development in Drosophila, which had been a central focus of his research ever since. His research group has made significant strides in elucidating the mechanisms undergoing organogenesis and growth regulation during embryonic development. Professor Shashidhara's research group has used the fruit fly Drosophila as a model system to discover gene regulatory mechanisms by which cells, tissues, and organs are positioned in their respective places in our body, how organ size and shape can be specified, and the diversification of wing number and size across major insect species. Professor Shashidhara has been instrumental in establishing both the Indian Cancer Genome Atlas and the Pune Knowledge Cluster, which serve as platforms for collaborative cancer research and multidisciplinary efforts aimed at addressing health, water, environmental, and sustainable mobility issues. He served as the Vice President of the Indian National Academy, Science Academy, and Secretary General of the International Union of Biological Sciences. He's currently the President of the IUBS, the first Indian to be elected in its 100 years of history. He had been awarded the prestigious Shanti Swarup Bhatnagar Prize for Science and Technology and the J.C. Bose National Fellowship, and has been elected as an MO member. In addition to his significant contributions to research, Professor Shashidhara has also been involved in promoting science education. He worked with the Ministry of Science Education on training high school and undergraduate teachers, in adopting inquiry-based teaching methods, organizing several career workshops for scientists in science journalism, policy, climate education, administration, and has served on the board of India Bioscience, reflecting his commitment to the life science research community in India. His work in translational cancer research is particularly notable. Within 3 years of shifting to clinical breast research from Drosophila biology, he has helped to establish a productive translational cancer research center. During the COVID-19 pandemic, Professor Shashidhara also had been working actively to mitigate its impact in Pune and across this country, including epidemiological data collection, analysis, serosurveys. Let us join our hands to give a warm welcome to Professor L.S. Shashidhara to share his journey with us and inspire young minds today with his experience. Over to you, sir.
Thank you very much, Dr. Jyoti, Dr. Ravindra, Dr. Uttara, for the invitation and also kind long introduction. I don't know from where you picked up all those. It's a pleasure to be here, and some of the colleagues here are known to me from earlier days too. It's nice to be here. My first visit to this campus, although I've been in the Manipal campus earlier. And a warm welcome to all the students who are joining today, both masters and PhD students in the area of regenerative biology. So I picked up today's this talk, given that you're all working in the area of, going to work in the area of regenerative biology, particularly students, and of course the faculty are already working in this area, which is truly interdisciplinary. I'm sure many of you, the faculty may not have worked in the area of stem cell biology or regenerative medicine, but they can contribute directly and indirectly because of their expertise in other aspects of biology and of course in general science, including data science. And so the topic, actually it's supposed to be a trunk twister some time ago. I just reduced the twisting part. It's supposed to be from science of life to more science for life, right? So the students here who are coming to understand regenerative medicine are the students of science for life. Now, without understanding science of life, obviously you will not be able to come out with applications or solutions for life. Now, when you say for life, there are three aspects of life. One is human life, because we are all humans, we look at everything with a very human-centric way. So our health, our food, our nutrition, our well-being is what we look at as the solutions that science should provide for life. Second is, if you remove humans or you consider humans as simply another animal on planet, then the entire environment, biodiversity, everything becomes part of our responsibility. Then again, we think about how we use science for life. So when you say science of life, can one of the students tell me a single word that explains all aspects of science of life? A single word that you think can explain. I'm sure it will be very obvious when you go forward. Evolution, right? Nothing makes sense anywhere in biology or life. Biology is what is there in the textbook. It's a technical term to study of the living systems, right? But life encompasses much beyond biology, beyond what is there in the textbook. In the textbook, you basically study anatomy, biochemistry, molecular biology, and so forth, right? Or physiology. But when you talk about life, life can be anything. Standing here and giving a lecture is also part of life. What happens in the family is life. What happens in the society is life. What happens in politics is life. How society is run is life, right? So even everything can be explained with the help of science, and that science that explains all aspects of life is evolution. So without understanding evolution, obviously, we'll not be able to manage anything in life. So for example, you take regenerative medicine. First you have to ask question is, why organisms repair and regenerate the tissues, right? How comes later? The question is why. Whether not all organisms do this. For example, many insects, they don't have any regenerative capacity. The Drosophila is a wonderful model system that many of us work. It doesn't have any regenerative capability. So why is that humans have? Why do that many mammals have? Most all mammals have. Many invertebrates too have, right? Vertebrates have, invertebrates have. So you can ask this question, why first? Because that's important. Without understanding the why question, you'll not be able to give right, sustainable answer. Many times we provide some answers, but those answers will be only for that particular time it solve the problem for that minute. It's like painkiller. You can reduce the pain, but you're not going to cure anything. So if you want to cure something, obviously you need to understand the disease, or you understand the wound, you understand the injury, and so forth. So in that context, you need to understand the why question, which the evolutionary biology will help you to understand that. So, pardon me, all the students, particularly the senior students and the faculty here, this talk is going to be very, very basic. I'm not going to talk about anything new. I'll talk in three parts. One part is about some aspect of what we know so far about life. There is much more we know than what we can do in half an hour of a talk or 45 minutes. The second is something about my own work, what I'm doing and what I've been doing in the last 3 decades or so. And the last part is some few slides about your own journey in science, particularly for students, most masters and PhD students. None of what I'm going to say is new to any of you. Even if you have not studied biology after 10th, It's not going to be new because everything I'm going to talk about in the first 20 minutes is going to be mostly the school, information. So first is about science. Science is the pursuit of knowledge beyond our own perception. It's something very, very important because all of us, we see, we hear, we learn, with the help of these 5 sensory organs, and we try to understand the world. And if you have some doubts, you talk to other people. But those people also have understood the world with the help of those 5 sensory organs, right? If you ask 7 billion people, right, if you have not studied anything in the textbook, what do they understand about the relation between the sun and the earth? They think that sun is moving around us, right? And we'll never be able to realize that earth itself is moving around the sun, and also earth is rotating on its own axis at very high speed, right? Because our perception, our ability to perceive is not enough to understand the reality of the world. What science has provided the answer is beyond anybody's imagination, beyond anybody's intuition, right? That's what science is about. You have to try to understand the world, surroundings, natural world, human world, beyond earth, which is going to be beyond anybody's perceived world understanding, right? And that we ought to call as somewhat absolute truth. And that's what science is all about. Science, methods of science is more important. Science is not physics, chemistry, math, biology as a content, what you study in the textbooks, what you study in schools and colleges. Science is a way of understanding the world, a way of understanding our surroundings. It could be understanding your own life, you're understanding your own brain, your own mind, understanding your own family. But that methodology that you use is more important, right? Scientific methods are 2 types, mostly, always be following what's called observational studies. And the second one is experimental, pre-designed studies, right? Now, observational is very, very old. Since humans evolved with language ability when they can talk to each other and try to express themselves with the help of language, observation studies have been going on more than 200,000 or even beyond. Whereas pre-designed studies, so-called experimental studies, much more recent. It's about 2,000, 2,500 old, started with Greek experimentalists. Now, if you want to ask the next question with the help of scientific methods, now what is life? Which is a very, very complex question, right? Obviously, there is, even today, we're still grappling with the right answer. Is there one definition that you can give to life? What is life? But intuitively, even a child would know the difference between life and non-life. If you show a piece of a colorful, crystal, and you show a worm, or you show a, coral, or you show a flower, a child will also be able to tell, what is living system, what is non-living system. But you try to understand what is life, it's still, a lot of difficulty while we are explaining it. But the modern science, particularly in the last 100 years, has given tremendous explanation to understand what is life, even if there is no one particular definition to what is life. But sufficient understanding to understand ourselves, understand life in general, understand ourselves as human beings, understand ourselves as an individual, me as a self, or aware, an organism who understands self, right? Because irrespective of what you talk about the world, about society, about your neighbors, your friends sitting next to you now in this auditorium, you have something called your own self. You must be thinking for yourself many, many things. That is also part of the life. And that's the most complex phenomena that we need to understand, what's called cognition and consciousness. So there are multiple different ways people are trying to address what is life. Started with Mendel. My story starts with Mendel. And again, no need to introduce what Mendel did and how he discovered laws of inheritance and so forth. Most important question Mendel was asking about not how we inherit very different traits. Traits could be simply a few morphologically distinguishable traits that he used, right, using the pea plant as a model system. But look at the fundamental question he was asking. But every time a new individual is born, whether the pea plant or a human being, first is a life is born. Then life is a particular species-specific life is born, and then some family-specific features are inherited. So while Mendel may have studied the third part of inheritance, but he was trying to answer what is that which helps us to inherit the life, all traits that constitute life. Because every time a human baby is born, it's life. Every time a pea plant is born, it is life, right? And so he came out with certain laws of inheritance. That means there is a fixed pattern. When you say law, means it's a fixed pattern. And we keep saying that there is always non-Mendelian inheritance, there is some exceptions and all those things, but they're all very, very minor detail. But there is a fundamental inheritance patterns which is, fixed and which is universal across all organisms. And that also sort of suggested that there must be something common across all organisms. Around the same time, although not known to each other, Darwin was trying to understand life by comparing different living forms. There was some extinct, so by the time we knew the fossil records, and there are some species had already become extinct, many, many species, of course. Those days, they thought that only few species were extinct. And at the same time, he also trying to sort of look at the comparison of existing species and how species evolve, right? How species come about on Earth. Use the word evolve, it gives an explanation, but if you don't know the mechanism, you simply ask the question, how did they come about on Earth? Did they all came on Earth at the same time, in the same form in which we see today? Or it's sort of a process in which change over time, different forms have come about. And that process of change, he used the word evolution. And his very simple explanation is, every time organism born, they are not identical to their parents, they're not identical to their siblings. You just look at yourself, none of you are identical twins to your own parents, none of you are identical twins unless there are monozygotic twins to your own siblings, brothers and sisters. And that's the variations is always generated in the population. Many times you have certain features which are not there in any of your family members because some new features are introduced because of some new variations that are introduced every time organisms reproduce. It's very easy to study using small insect like Drosophila because you can compare thousands of flies in a small population, leading to a larger population a few days later. Or you look at one particular pair, one male and one female, typically you can get about 200 flies, and you can look at all the 200 flies and see the variation in those 200 flies. Although just coming from one single female mated with one single male, you can still see so much of variations. These variations, when they are opposed to environment is constantly changing, which is very obvious, is intuitive and commonsensical. The two kinds of environment, one is biological environment, one is non-biological environment. Both are continuously changing. And also biological activity will change the environment. For example, oxygen in the atmosphere is because of biological activity. Before photosynthesis, there was no oxygen in the atmosphere, right? So given this, the environment is constantly changing. Some may be able to adapt, some will not be able to adapt. What happens is when the variations is more in the population, certain variants who are able to adapt to the environment will reproduce a higher frequency, some will reproduce a lower frequency, and this will change the population structure, and over the time, it may change to such an extent that we consider those two populations are two different species. But they're all in one continuum. If you look at the tree of life, it's just one continuum. If you take one line, mathematically, 0 to 1, 0, 1, 2, 3, 4, up to 100. Between 0 and 1, you have 0.1, 0.2, 0.2, whatever it is, right? You have these numbers, values you add per position. The two different groups which are closer to each other with similar values, obviously they will looks very similar to each other. And two distant ones, they look so different, you think that they are two different, completely different groups, completely different types of organisms, they are not connected to each other. But still they will be standing on the same line. But if your line is not visible to you, you consider them as two different individuals, right? So in that context, for example, look at yourself. Your brothers and sister. Someone who comes to your house, they'll assume that you must be brother and sister because there are some similarities. If brothers, sisters, and cousins are together, they know there are some similarities, but they also know there are differences. These two individuals who must be somewhere different because they are not very similar to these brothers, sisters, they think that perhaps they are related, but not necessarily in the same family, right? So if you go to a marriage where there are hundreds of people from the family coming, first cousin, second cousin, and all kinds of people, they will clearly make out who could be belonging to the originally that particular family, who may not be. We may not be able to identify the relationship, but at least you will be able to grossly make out. But if you go to a conference like this, people come from all kinds of families, all kinds of backgrounds, all kinds from geoclimatic conditions, from different parts of the country and so forth. Obviously, it's very difficult to look at. But you all can find out that they're all human beings. Similarly, if you go to a UN summit in New York, there will be almost all 250 countries are represented. You can make out they're all human beings, but they're so different from each other. But tomorrow, if you expand our United Summit to also include chimpanzees and gorillas, who are our first cousins, so now you see that they will look very different. They may not be from this planet. They may not be anything connected to us, but they are still part of the same line, somewhere a distinct dot which is slightly away from us, right? But if you compare a Drosophila and human, that dot is far away from us. But for a Drosophila, for human and chimpanzees are identical, right? Because they are so close to each other. So this is how Darwin, perceived the understanding of life. And he mentioned, how very different forms may evolve on Earth. Though both he understood the mechanism and the reason why we have such biodiversity on Earth. But still, the mechanism was not sufficient to give a clear explanation, because his understanding of mechanism is still early at the level of variations are generated and they are opposed to environment, and then there is what's called adaptation. Now, understanding of evolution, which is very important for the students here, why do we use model organisms? You may ask the question, why can't we use directly humans? Because whatever is studied in model organisms, will it be applicable to humans? So understanding of genetics and understanding of evolution, and then there was a convergence happened in the beginning of 20th century. People realized that we should be able to study most aspects of life using one or two model organisms and then try to extrapolate to all other organisms. Because if each one of you working on different different organisms, they are not necessarily model organisms, you may scratch the surface about understanding what is life, but you'll not be able to get deeper understanding. You will not be able to get a sufficiently good understanding of life where you can use it for certain applications, whether in food, agriculture, or health, and so forth. That's why people decided, let's use certain model organisms. And now, for example, Drosophila is one of those. There are many other, zebrafish, C. elegans, Arabidopsis in plants, and so forth. But thousands and thousands of people have been working on this for the last 3 generations, more than 100 years. And cumulative information that we have generated using fly as a model, it is so much that we can actually get a deeper understanding of what is life, and that can be extrapolated to all other organisms, including human. Not only we understand the development, genetics, evolution, biology, molecular biology, biochemistry, physiology, neurobiology, aging, and also some of the human diseases, and also, of course, understanding the behavior, cognition, and also, flies do have what called self-awareness in some kind, in their own way, and you can understand even the complex, behavioral outcomes too. Around the same time, microscopy, which was there for last 250 or so years, people were trying to understand what is constitutions of life, right? So people thought that if you understand some aspect of genotype without knowing what is gene. For example, genotype and phenotype can be explained without knowing what is gene, right? Molecular level. So genotype-phenotype correlation, there must be some mechanism of which genotype manifests as a phenotype. Phenotype is something what you see. And that's where the cell biology sort of started emerging as a strong field to understand something must be happening in a cell which basically leads to what's called phenotype. Color of the skin to eye to very different things that morphologically you can look at from outside, or something which is internally, for example, resistant to temperature, or plants, for example, how do they resist water scarcity or salinity in water and so forth. The same then, understanding of genetics is a fixed pattern. People realize that must be some material basis for life. It cannot be an abstract phenomenon. It can't be something very elusive, material basis for life. And that's when, because there's a fixed pattern, right? Every organism's, the patterns of genetics, pattern of evolution that you could experimentally prove. By 1920, people had already done a lot of experiments, both in evolution and in genetics, across different organisms and populations. So they started looking for material basis of life, and that's where they started looking at inside the cell. And they found out there is a chromosome, the chromosomes are the carriers of gene, and later found out that chromosomes themselves must be the genes, and then they're not just, the genes are nothing but, some units, and that units are, arranged on this chromosome, and all kinds of cytogenetic phenomena was there. Continuously, every decade, in those days, very exciting time, new concepts are evolving. And people could actually, using Drosophila as a model system where you can see these large chromosomes, there are certain specific cells in which these large chromosomes are present. Under a microscope, you can actually see very, very fine details of the chromosomal one. For example, you can, if you can see my cursor here, for example, the tip of this chromosome, this is the X chromosome. A tip of X chromosome, morphologically very different if the fly has a red eye color versus white eye color. So that means there must be, very excitement if you look at, there is a clear material basis for characters such as eye color. So they understood that we should be able to understand life if you understand the structure of what is there in the chromosomes. And they found out that chromosomes are made up of certain kinds of chemicals that is called nucleic acid. Those nucleic acids are 2 types, and chromosomes carry mostly the one type of nucleic acid called DNA. And I don't need to explain to this audience. It all fast track if you take it 50 years. We know now DNA is the polymer which stores all the information about very different aspects of life, and how DNA makes copies of itself is fantastic phenomena that we need to understand if you understand the genetics, if you understand how variations are generated, how variations spread in the population. All that can be mechanistically explained at the molecular level by understanding DNA replication. Most of you know what is PCR and RT-PCR. PCR is nothing but a DNA replication process that happens outside the cell. Now, DNA, how the information is decoded in the form of RNA, and then from RNA, how the proteins are made. These proteins are the real workhorses in the cell, and everything, the structure of the cell, the physiology of the cell, and the biochemistry of the cell is based on the protein functions, and that information is generated. There are a lot of variations in a lot of flexibility, robustness, all that thing we understand about life can be explained with the help of how DNA information is transcribed and translated, and how protein function once they are made functionally, functional proteins are made in the cell. So that's where biochemistry and physiology connect, right? Now, at the same time, before I talk about physiology, which is very important for all of you, particularly those who work in regenerative medicine, people realized by 1950s, when they started seeing cells, this is where the non-biologists, biologists to some extent, those who use microscope already knew that cell itself has all the features of life. It's not that cell is simply a building block of life. It's not like a brick which builds a building. Cell itself is life. So all multicellular organism is simply just a collection of large number of cells. For example, human body has about 50 trillion cells. It's simply a collection of large number of living cells. So you take a drop of blood and see under a microscope, what is moving is neutrophil, and neutrophil is chasing bacteria here. And it knows where to go. It is what it's targeting. It's targeting a bacteria. It keep making its way in the middle of all these RBCs and trying to sort of capture this bacteria. At some point, it will engulf that bacteria. You can make out if the bacteria goes inside the neutrophil, suggesting that these cells, although they are part of a multicellular organism, they are independent to them large extent, and every cell has certain independence, and each one of them are functioning in their own way. And now we know that all cells they have their own metabolism. There is nothing like a centralized metabolism that some energy is produced in one part of the body and supplied to the whole body. It's not like one, electrical, let's say a nuclear reactor producing electricity and supplying to the whole city. It's not that way. Every cell has its own tiny mitochondria, a large number of mitochondria, not just one. These tiny mitochondria in the cell, and they generate energy for what happens in the cell. So when you have to move a body like ours or a body like an elephant, large number of cells have to work together in synchrony. And that's whole, it changed the whole understanding of life when you say that life is all about synchronization of all these biochemical reactions. Now, biochemical reactions are like, ATP synthesis is a biochemical reaction. Photosynthesis is a biochemical reaction. Now, if multiple biochemical reactions are aligned in time and space, and they are connected to each other, we call it as physiological reaction. In a way, actually, photosynthesis is a physiological reaction, not a biochemical reaction. When photon hits the first protein, in the photosystem 2, an electron jumps, it's a biochemical reaction. But electron going to next protein and that protein getting activated, and then the splitting water this requires about 4 or 5 different reactions. All of them together is a physiological reaction. So finally, we now understand that life is all about a series of biochemical reactions happening in sequence, in highly regulated way, in time and space, and multiple cells, particularly multicell organisms like ours, they work in synchrony. For example, those who want to understand diabetes, there are type 2 diabetes is not about loss of insulin. Type 2 diabetes is complete loss of insulin synthesis. Type 2 diabetes is actually reduced levels of insulin, which leads to complications later, like our AMR, right? Emergence of and insulin resistance happens because of irregular secretion of insulin. It is a loss of synchrony in insulin secretion which leads to type 2 diabetes. So today, if you ask what is life, we still do not have one sentence to explain what is life, but at least we have a small paragraph which can explain all aspects of life. What is written in the red is most important aspect of life. Without variations, we will not be able to survive because the environment is constantly changing. We are changing the environment. Environment itself is changing because of very different universal features of the stars and galaxies and so forth. And in the process, if no variations are generated, we'll not be able to adapt. If you don't adapt, obviously you will die. The fact that we have life on Earth for the last 3.75 billion years is whenever the first form of life originated, it had the ability to change, and that change is what led to adaptation to varying environment. And that change also we know now. The copying mechanism, all of you know, it's the, two complementary strands come together because of base pair matching. The guanosine and cytosine make 3 hydrogen pairs, and thymidine and adenine makes 2 hydrogen bonds, and these are the perfect Watson and Crick pairing. Sometimes they can actually mispair, and that mispair is what is the genesis of new variations. That can happen very rarely because the lots of mechanisms have evolved over time to reduce the number of such mispairs by proofreading mechanism, variety of different other DNA repairing mechanism also. But still these small errors, we call them errors, but they're not really errors, variations are introduced. Those variations are the fuel for evolution, right? It happens very rarely, it's actually 1 in million, but the 1 in million in a genome of a human, for example, haploid genome of human is 3 billion base pairs. So every time, for example, I have one daughter and I must have passed on at least 3,000 new variants to my daughter when I passed on my genome. Similarly, my wife must have passed on another 3,000. So my daughter would have collectively 6,000 new variants in her genome, which is not there in either of the two parents, right? And because of the semi-conservative nature of this replication, whenever there is a variation is introduced, that gets fixed in the population. The next time when the DNA replication happens, then it can have a normal pairing. For one time, it will be a mispair. The next time it will become a normal pair because the G will go with C and A will go with T. Even if it's one time, let's say G happens to go with T or A happens to go with C. Then we need to understand how structure evolves and function emerges. What evolves is a structure. There could be a one base pair difference or 2 base pair difference in DNA leading to some changes in the protein, and protein is a structural element. Protein has certain function. But finally, what we call as function, how does it evolve? For example, you can always say the arm, the structure like arm is evolving with time, but how the function of arm would emerge, right? For example, something, a flat structure comes here, will it become a wing? When bats, it has become wing. So what is that property of emergence of the function? That's where evolution plays a big role, the mechanism of evolution. So, for example, they take this example. Why in a particular function, if I had a certain benefit to the fitness to the organism, it will go in the population. Sometimes we think that every variations which is not part of the today's function is actually lost to the fitness and you should not get selected in the next generation. Look at this example. I'm sure the students of this particular institute should know the sickle cell anemia. It is caused because of one amino acid change in hemoglobin in the 7th position, because of one nucleotide change in the corresponding gene, leading to making the protein which is somewhat different structurally, and it is packed, it's not so nicely the way regular disc-shaped RBC is here in the top, and what you get is a sickle cell shaped RBC. But interestingly, this, while that sickle cell RBCs has certain deficiency in oxygen exchange, heterozygous individuals at a higher altitude do have anemic phenotype, but at the mean sea level they are perfectly normal. Let's say if also malarial parasite is present in the environment, and malarial parasite has evolved, is co-evolved with human to infect obviously the regular disc-shaped RBC, because that's in the majority of the population. So malarial parasite, it cannot enter sickle cell shaped RBC, because of which they are resistant to malaria. So in a mean sea level, if it's endemic to malaria, in fact, sickle cell individuals are better off compared to so-called normal. So there's nothing like normal, abnormal in the population. Everybody is different. Depending on the context, depending on the environment, some have a better ability to survive and reproduce, some have less ability and survive and reproduce. So that's what functional emergence is all about, which is context specific, which is the environment which decides. And this is going on for 3.75 billion years, and that's what we see today. This is our National Geographic picture of biodiversity, and you could go to any of the forest in India or everywhere, in Western Ghats or some other place, you will see really fantastic biodiversity, whatever little we have retained with the human activity because of destruction of the environment, but still it's a fantastic picture to see. At the same time, we also know now, I'll not get into the details. At some point you can understand that we have sufficient understanding that must be a common origin of life, because the very chemistry of life suggests that must be a common origin of life, because from across all system, exactly the same chemistry of DNA. All your recombinant DNA technology, talk about it in a minute later, is possible because of the common chemistry of life, right? And so obviously there's also common origin. Then the question comes, chicken versus egg. For example, how does this itself is a very complex phenomena, right? Forget about cells, forget about multicells, everything. Simply an organization where DNA, RNA, and proteins are products of each other, and then how it happens. DNA itself needs to be replicated, and that requires proteins. RNA transcription requires protein, and protein synthesis requires proteins. Then what is the beginning if, from all of these things? So we have some understanding of this, some experimental evidence available, some hypothetical, some theoretical, some mathematical. People are working on it. So application, you have students of working on science for life. Earlier days it used to take time, but now it's also fast-tracked. So I'll just talk about two examples. One is, again, using insulin as an example. So Darwin proposed his hypothesis in 1859, which became more or less a theory in the beginning of 20th century, with so many people verifying, validating his hypothesis. And by 1920, people realized that there is something called protein, some X protein, we call it insulin, is required for glucose metabolism. If that protein is not functional, if protein is not enough, and glucose level goes up in the blood, which we call as diabetes. Now, until 1920, there was absolutely no treatment for diabetes. You somehow control the sugar levels. People knew that there is something related to sweetness. That's why in Kannada or in Sanskrit they call madhumeha for diabetes, because the urine from those patients attracts ants, and that's how they actually diagnosing for diabetes in centuries old earlier. But irrespective of their affordability, there was no really treatment for diabetes. But with better understanding of evolution and biochemistry at that time, people realized that if you extract insulin from other mammals, we should be able to treat diabetic patients. So we extracted insulin from slaughterhouses, from pigs and cattle and horses, and they were trying to lyophilize that and send it to hospitals, and hospitals were basically giving this as a treatment to diabetic patients. But because you extract very small amount of proteins from large number of these animals, and then it became very expensive. So for some decades, diabetes was considered a rich person's disease because treatment was very, very, very expensive. But look at, but better understanding of life, particularly molecular biology of life or biochemistry of life, now we can make insulin in small cells, bacteria or yeast cells, because the chemistry of life is identical. You take DNA of human and put it in bacteria. For human, when it is transcribed and translated, what you come out with a protein which can control glucose metabolism. But in bacteria, there may not be requirement for such a protein. Bacteria can still produce a protein, insulin-like protein or insulin itself, for all practical purposes, because bacteria will see a piece of DNA which will undergo replication in bacterial cell, it will undergo transcription and translation. Finally, what comes out is insulin protein. Now in this kind of hall, you can make kilogram and kilogram quantities of protein, and now insulin is so cheap in almost in all government hospitals across the world, they are actually given free of cost to poor people. So they're absolutely social equity now in terms of diabetes treatment because of this remarkable understanding of life. And today, this took about 100 years, because even if whatever little we knew is not enough for application, we need to discover more, and finally it happened. Look at this, today's how we address the pandemic. Your registrar talked about building this whole campus during the pandemic time, and it's a remarkable achievement. Not everybody could do it. But pandemic, we could sort out, because all these could manage. We could manage, although there was a severe lockdown for 3 months, but beyond all of these things, we could manage many things, including education, although there was inequity in how people could access education in the society, because not everybody had the gadgets or everybody had the space to learn online. But at least we could somehow manage the life on Earth, because we knew how to detect virus using RT-PCR. Otherwise, how do you know whether there's virus in the air, virus in my body, right? We could use RT-PCR methods. We could develop vaccine. All this happened within 1 year, right? By end of 2020, already vaccine was rolled out. Although India, started vaccination in massive only after April 2021, but typically by December, US and UK already had rolled out vaccine for public use. So that's the, how we can, fast track now because of such deeper understanding of life. In India too, we have used science to alleviate hunger and poverty. There are multiple examples of how India has used science to address societal problems. In the context of biology, I'm talking about a green revolution. Everybody know milk revolution. Everybody know how we have used science to address problem of food and nutrition. Because when we started in 1947, we are a country of one of the poorest of the poor, with absolutely no agricultural production enough to feed our own people. So we had to and we didn't have enough money to buy food. So obviously people were donating food to us. From that, now we are exporting, we are donating food to other countries, right? That happened because of use of science and technology in building the nation since 1947. And we use genetics and evolution, which helps self-sufficient food and nutrition. We use mathematics and physics. We know India is very strong in mathematics and physics, traditionally also. And that helped in building our atomic research, our space research, our IT industry. All that, Bengaluru is all about IT industry, is because of the strong basis for physics and mathematics in this city and in the country. Chemistry and chemical engineering made India the pharmaceutical hub for the whole world. And now molecular biology and biotechnology has made large biopharma. Now India is also the largest producer of vaccine and supplier to the world. But there are also some called innovation stories, for individuals. They're all collectively what we have done as a nation. If you look at the individual level, the very discovery of cholera toxin, right? It's a remarkable example of process of science, how systematically one was studying and what causes the disease cholera because of infection, right? A mycobacterium infection. Sorry, not mycobacterium. Vibrio cholera infection, right? Then another story is about how iodine as a supplement in the nutrition is required to reduce very different types of, pediatric problems like goiter and other, abnormalities. And work of Shamunath Dey, Dilip Mahalanobis, and Professor Ramalinga Swami, they have actually saved millions and millions of children from in India, Southeast Asia, in Latin America, and all those places. Because of, there are two reasons. Poorer countries are the ones where the nutrition is a major problem, and particularly the small, minor, so-called minor, constitutions of the nutrition that is required, like vitamins and minerals and so forth. The second is, climatically also, some of these places do not have sufficient those nutrition content in the food itself. Whatever the food that we produce, for example, vitamin B12 deficiency is not whether you're vegetarian, non-vegetarian, you're still poor in vitamin B12 in India, is because of very geoclimatic condition in this country. We don't produce enough vitamin B12 which we can absorb in our body. Even if you take these tablets, is not good enough because absorbance of vitamin B12 these tablets is not good enough. You need some biological intervention for that. Then the research is progressing. For you students, your world is different. It's going to be different further. It's already very different compared to our world. I don't think ever. And how many of you have stood in the queue to get some ration, rice, or oil, kerosene? And I don't think any of you students, I'm sure my age people, we all used to stand for hours in the queue for these kind of things. The world has changed so much, but more than that, economic change in a positive way, world is also changing in a negative way, variety of different way. One positive aspect of today's world is we are all connected. We are connected virtually, but and in real time we can exchange information, which is very, very important. At the same time, there's also negative change, huge negative change. We just, every day we are hearing, what happened in Wayanad, what's happening in Uttarakhand, the cloud burst, Himachal, Pradesh and other places. And, the floods, the droughts, everything has become more and more frequent. And our resilience to sustain some of these catastrophes are also coming down. 300 plus people have died in Wayanad just last 3 days, right? But one of the features of climate change is, while climate change or global warming is global, right? But solutions had to be local because the impact is different in different geoclimatic situations. So it's not like somewhere in Boston or somewhere in Stanford, someone will discover something or invent something which is going to solve the problem of the world. We cannot rely on that. Every place had to innovate for itself. And that's where science has to go to the grassroots level, to every village, every town, every city, in every country all over the world. Then only we can move to sustainable living, we can achieve the sustainable living goals. Otherwise, there will be only a commercial product helping the climate change, but that's not going to be useful every part of the world. Now, your world is so different, and how do you predict what kind of jobs you will have? Today we are talking about artificial intelligence doing many, many things that we, 10 years ago we couldn't do it. 10 years we were thinking that so many things computers are doing, very different apps are doing. 20 years earlier there was our call centers, very different IT services, those are gone. So even in one sector, IT sector or agriculture sector, there's so many changes happening. Then you prepare yourself today that you will become an expert in regenerative medicine. What kind of skills you need to be able to provide solutions, whether you're in a medical hospital or in a college like this or in a biotech industry, right? It's still very difficult to know what skills would be required. So better to learn, acquire transferable skills. Transferable skills are something which help you to prepare yourself irrespective of the environment in which you're going to live 5 years from now or 20 years from now or 10 years from now. So one is of course ability, then innovation, ways of connecting knowledge to the problem, real life problem is called innovation in single word, but there's a long sentence for this. Critical thinking and self-learning ability. So they can learn quickly. All of these things come naturally if you do research. So places like Mahe, where research and teaching integrate, where all your faculty are researchers, they have come from high quality research experience, and they are going to teach you because of their research experience. The teaching method will be different. Their laboratory level, there will be nothing like demonstration. You're going to do your own experiments. You're all going to do small projects with your faculty here. And then you will have particular PhD students, of course, all about research. Even masters will be doing some master's thesis research. If you do research honestly and with integrity, all of these things you will learn. Because you cannot do research without these abilities. If these abilities are acquired, rest of your life is taken care of, irrespective of what kind of world you're going to live your time. Most important aspect of, I said, if you do it with sincerity. The sincerity comes as a philosophical underpinning of science itself. Now, if you look at what is science, you make an hypothesis, you test an hypothesis, validate your hypothesis, verify your hypothesis. Why do you do this? Because you consider hypothesis wrong. Science is the only place where you call yourself, perhaps you may be wrong, you need to verify yourself. Look at all others, historians, philosophers, religious gurus, politicians, right? In an economist, they have certain firm belief in their own understanding of some hypothesis, that nothing like hypothesis, they consider that there is a truth. And they keep on saying that there's the only truth, there's no other truth. And you have to disprove them. They say, you disprove me, otherwise, I'm correct. And then you go on say, try to, argument, trying to disprove them. They keep on saying that, no, I'm correct, it's not good evidence. If you say, for example, don't take me wrong, if I say, there's no God, a religious person say, show me how there's no God. You ask them, you challenge them, you show me where there is God. This argument goes on forever, but finally the religious person will continuously say there is God, right? So if you, in science, is the only place where you say your hypothesis is wrong and all your experiments are designed to disprove your hypothesis, right? If you look at, carefully, those who have done some projects, you use what is called null hypothesis. Null hypothesis is the opposite of your hypothesis, right? And all your p-value is to show why null hypothesis is true. So, for example, p-value is 0.05, you say. What it means is, it's a 5% chances that null hypothesis is correct. Because null hypothesis is correct only at 0.05 probability, that means 0.95 probability is the reverse of the null hypothesis is correct. So, you assume that your hypothesis is correct. Only if p-value is below 0.05 or something, right? Depending on 0.01, sometimes physicists use 0.0001 p-value, right? Whereas biologists typically, behavioral biologists say 10% is also good enough, right? So that means you start with a very obvious question that yours are wrong and you want to prove yourself wrong, not that you want to prove yourself correct. That itself is a very interesting, philosophy of science. And because of which you actually follow a certain, over the time with 100 years, we have followed what is called scientific methods, research methods, which is extremely based, foundational on ethics and integrity. Data fabrication, manipulation, we see that people will see retractions, there are statistical tests they manipulate, but every one of them has emerged to actually, as a scientific methods, to prove you wrong, not prove you correct. If you try to use all of these things to prove you're correct, you're actually not honest. Science is based on honesty and integrity, and without which you cannot do good science. So again, students of biology here have a strong foundation in genetics and evolution. You cannot do regenerative medicine without that. Biochemistry, cell and molecular biology, genomics, and physiology. Make sure that you have good computational skills, because modern world is all about data science, and you can do large data integration, interdisciplinary work you can do with your computational skill. Have good communication skills, not 140 character communication. Long writing is very, very important. A lot of misunderstanding can happen if you try to communicate in short words or few phrases. Have long writing skills. Research skills I already talked about. Entrepreneurial skills. Entrepreneurial skills is not about setting up your own startup or do business. For managing your own research is entrepreneurship. Right? Managing your own research. In the lab also you need to manage your own research. So your time management, your resource management, and other people management, they're all part of the entrepreneurial skills. Okay. How much time, you have to ask me how much time I can spare, right? When did I start? 10:30? 10:20, okay. So almost another 5 minutes I'll come. So just quickly, because I'm standing in front of you, are all biologists, you're all students of genetics, biology, molecular biology, and cell biology, I should brag a little bit about myself, right? So my own work. Started as a developmental biologist. My understanding of development is about starting from a single cell. How do you get this complex organism? In this case, for example, human baby is born is about 50 trillion cells, and that's constitute the major part of the body. There are a large number of several more trillion cells who are called stem cells. I'm sure you know what stem cells are. And then, of course, several trillion bacterial cells which constitute the microbiome. Finally, a human is a combination of differentiated cells, trillions of differentiated cells, trillions of stem cells, and trillions of bacterial cells, right? Then without that, you cannot call someone as a human being. And so the question you can ask is, how do positional information generated? Why do you have eyes only here, arms here, and legs here, and kind of thing? And how do we get perfect size and shape proportionate to our body size given a particular species? Drosophila has a particular species, a particular body size, and proportionate to the body size, there are certain kinds of wings and legs. And how do these size and shape are determined during development? Because every time you start with a single cell, there's no predetermined everything. Then the physiological connection, because otherwise physiological connection, you cannot work as single, because I told you, every cell itself is a life. Then if your trillion cells are coming together, how do they come together as one life? If I'm lifting my hand when giving talks, billions of cells will be going up and down. So what is that manage, the synchrony is managed? That's because of the physiological connection. At the mechanistic level, because you start with single cell, given human cell, there are certain cellular features, and those cellular features are common across all trillions of cells, because all trillion cells come from the same cell through mitotic cell division. So obviously some improvised basic cellular mechanism to make something muscle, something neuron, something, a cardiac cell or a liver cell, and so forth. Second is complex, particularly in the context of size and shape. Every cell, even if like yeast, unicellular, free-living organism, has its own cell cycle regulation. When to divide is extremely well controlled. And that, because of certain ways, it happens what you call as cell cycle regulation. A few people have to look at it. But there, if you have multiple cells have to divide in synchrony and then constitute to a particular tissue, then the cell division control has to be extremely well regulated, and it becomes much more complex, right? And finally, it all comes down to spatiotemporal gene regulation, because you start with the same genome, you get all kinds of very diverse cells, but genome remains the same. How do you, if you look at, if you ask anybody and show a picture of muscle and a picture of a neuron, and someone who doesn't know biology, they will think that they're coming from, they are two different unicellular organisms, must be something maybe marine organism, something maybe terrestrial. Maybe marine one is the one which is neuron, which has a long axon, which we use for swimming and so forth. But it's actually two different cells of our own body, starting from the same original single cell embryo or zygote. That means there must be some spatiotemporal gene regulation which leads to these kind of changes, because the genome remains the same. So the particular, these are the kind of questions I was asking in my lab using Drosophila as a model. And there is a particular family of proteins called Hox proteins, which are expressed in a specific, during the early development, they start expressing in a specific way. And depending on where they are expressed, you get different parts of the body along the anterior posterior axis, that is head to tail axis. So one particular protein I've been working on is called ultrabithorax. The transcription factor is a DNA binding protein. It's short is UBX. It is required for the specification of the third thorax segment. If you remove UBX, you get duplication of second thorax segment. There are two second thorax segment. There are 4 wings. Otherwise, the dipterence, all dipterence means there are 2 wings. And what is lost is pair of these small club-shaped organs called halteres. They are the balancing organs. And if you overexpress UBX in developing wing, you get 4 halteres. What this experiment, which won Nobel Prize for Ed Lewis and many, many other people contributed, suggests that UBX is required to make third thorax segment with positional information. It is required to make halter in that particular segment, which is specific tissue or organ, and it does so by modifying wing fate into halter fate. That means it specifies, very different size and shape of this particular tissue as against this, because if you move UBX, what you get is a wing. That means it's working on some ground state and modifying it. Then it is sufficient to make halter, because one protein is good enough to convert a wing to a halter. So it's a starting point. It's a fantastic genetic system where you can have two contrasting morphologically different organs starting from a similar progenitor. And now you can ask all kinds of questions about their positional information and so forth. So we asked two basic questions. One is UBX being a transcription, how does it identify the targets and changes the fate of wing to halter? And second one is what kind of growth and patterning events are regulated by UBX, because wing is a flat, large structure and halter is a bulbous, small structure. This is the kind of work we have done over the years. We also looked at, this is always my passion for evolution, look at from evolutionary perspective. If you look at all holometabolism, in fact, you must have heard the word metamorphosis, right? Metamorphosis is where in the same life cycle, there are two phases, but they're completely different looking individuals. The larva versus adult fly, or a caterpillar or a butterfly. Beautiful butterfly coming from this thorny caterpillar, right? And there are two different stages of the same organism. It's not two different organisms of a population, right? And that's because there is a particular stage during development called metamorphosis where this happens. There are only 5 groups of insects which go through full metamorphosis, and these are the 5, dragonflies, honeybees, and beetles, moths and butterflies and flies, fruit flies, house flies, and mosquitoes and forth. And each one of them have slightly different organizational structure, but only Drosophila has the third thoracic segment, complete absence of wings, no semblance of wing-like features. So how wing itself has two wing structure or halters have evolved during evolution is something which I've been addressing. Another question. I'm going to talk about the results or anything. It's just telling you what kind of work I do and why I'm doing those kind of work and what is the rationale, what is the scientific method we used. So we have, over the years, we have looked at very different regulatory pathways which are involved in positional information, which involved in growth control or size control and so forth, at single cell level and multicellular level and so forth. And many of them are also implicated in cancer because there are many of these, because cancer in human, of course, nothing in Drosophila, we can induce cancer in Drosophila in an artificial way, but there's nothing like cancer in Drosophila as a disease. The reason is the growth control is where the cell division control happens to limit the size and shape of an organ. And when that goes wrong, later in the stem cell, you get cancer. So many of the work that we did has implication for cancer. One of them is this inverse relation between cell size and cell number. So if you increase the cell number, typically cell size decreases. If you increase the cell size, cell number decreases. And this balance is maintained only for a certain range of cell number and certain range of cell size, which is very important to maintain organ size and shape, because cell size is generally determined based on the nutrition. The nutrition sensing pathway determines the size. Number is based on the cell proliferations control. And if the two are somewhere broken, this relationship, what you get is neoplastic growth. That's what cancer is all about, right? So just before I talk about cancer, why do you get cancer? Because we have stem cells. Why do we have stem cells? These are the evolutionary biology questions. There are certain advantages to have stem cells because you are a long-living organism and you live in multiple different environmental conditions. Because if it is an insect or a small ant, a worm or someone, which lives only for a few hours or in one location, it may not have all kinds of features to adapt to changing environment. Long-living organism, particularly highly mobile animals, every minute they may be in a different environment. So they need to understand the external world and try to adapt to the external world in their own living. I'm not talking about adaptation over generations, adaptation within the life period of an individual. So our brain helps to understand the world and adapt to changing world. At the same time, our body also has many, many features to adapt. Temperature resistance to very different physiological features of the body which help us to adapt to changing environment because we are continuously exposed to new environment as long as we are mobile. So stem cells have evolved in these kind of organisms to help live longer because with continuous use of heart or liver or very different lungs and everything, there is a wear and tear in the tissue. You need to repair that one, right? It's like using the same car for 100 years. You change the body sometime, you change the painting, you change the body, you change the wheels all regularly and all those things. At some point you may change the engine itself, and 100 years later what you see is a completely new car because so many changes happened. But it's same thing here too. Actually, we think that you are the same individual. Of course, because we have these neurons which live longer and that memory lasts for long. most of our body parts are actually continuously changed, but not all parts, like, some parts, right, is changed so much that finally what you have is basically like inner lining of gut. It changes every 2 weeks. More or less, what you're born with and what you die with will be a lot of differences in the organ system. That is because we have stem cell. The second role of stem cell is wound injury, because we are continuously exposed to all kinds of things. There can be scratches, there will be wounds, sometimes they're cut, some severe, sometimes small. You need to heal those wounds, otherwise they can be damaging to the function of the organ or the body whole or the life itself, and also because of infection, it may lead to more lethal injury. So wound healing helps to reduce all of these problems, including the infection problem. So stem cells are required for that. What are stem cells? Are predetermined but undifferentiated. They are division competent, but they are quiescent. They start dividing only when they need to be divided, right? So division signal come from, let's say, injured cell, or because of certain hormonal signal, or because of wear and tear happens in the body, and those trigger the signal to nearby stem cells to come and replace the worn out cells. But cancer is an unwanted outcome of this evolutionary mechanism which helped us to survive longer and healthy. Without stem cells, we cannot survive longer and healthy. But at the same time, because we have stem cells, there's a cost to this one, and the cost is incidence of cancer. Irrespective of whatever the different mechanism evolved to keep us well controlled without tumors or cancer, that happens. When stem cells start dividing without any control, it starts becoming tumor, and this tumor, multiple ways are suppressing the tumor in the biochemical, physiological, and genetic system. They keep on escaping something. It's like this, right? It's like an accident. More the number of events that you go through, there is a chance that something may happen, right? For example, Bangalore-Mysore highway, people say it's accident prone. And all of us keep traveling between Bangalore and Mysore, once in a month, once in a year, once in 2 weeks or 3 weeks, something. But if someone tries every day 3 times between Bangalore and Mysore, the probability that that person meets an accident is higher. It simply as a probability factor, right? And that's exactly what cancer is all about. And there are so many ways you can suppress cancer, but still we get cancer, is because of our population size, they live longer, and there are so many stem cells in the body. So I started working on cancer because of my understanding of development biology, growth control, evolutionary biology, and biochemistry and biophysiology. So now I'm working more on triple negative breast cancer. I'll skip all of these things and stop here. Thank you very much. You're waiting to stop me. You're waiting to stop me here. Okay, maybe I'll keep this slide just for...
Please take a seat first. We'll take 2 more minutes. Do we have time to take 2 questions?
Yeah, sure. That I can't say no.
So we would like 2 questions, one from the students, one from the faculty. Because of time constraints, we would like to minimize the questions before we end this session now. Any student who has a question for him? I know. I think we can have any one question from the audience. Okay. So I just want to make a note that I think the biggest take which I feel from this lecture as a researcher, I have taken that the key to unlock how to do interdisciplinary science or research is something which Professor Shashidhara has very beautifully explained using science as a tool and the various types of research that he has done all his life till now. I think that is something which the younger generation should really understand, that it doesn't really require you to belong to a particular niche field only for the entire life to work on. There can be definitely networking in between different fields of research. It's just the communication that you really need to understand how to interpret each other and work accordingly for the betterment of science and the community. So with this, I would like to kind of thank Professor Shashidhara for this wonderful talk, this honor lecture, and we give him a big hand. I would call upon Dr. Jyoti Prasanna to hand over the token of appreciation from MRM to Professor Shashidhara. Please give him a big applause. Thank you, sir. So, this session ends now, and we can break for refreshments for 15 minutes. We need to be back on time after the tea break, and we will begin with the next session. Thank you.
Motor.
Motorola Edge 50, crafted for the bold.
Hello Motor.
अगोड़ा पे पाइए बेस्ट होटल्स इन कमाल के प्राइसेस।
Domestic and international दोनों.
अगोड़ा।
It's nearly the afternoon session, but not yet, so I'll still call it good morning. Welcome all of you again for the second session of our orientation program. I want to call upon Dr. Jyoti to introduce our course at MIRM. Dr. Jyoti.
Thanks, Menal. Welcome to the magnificent Mahe, Bangalore campus. I don't know how many of you have gone around today morning. Have you gone around or you directly came to the AB4 auditorium? Okay. I think after all the talks are done, you should go around and see our facilities. We have a beautiful Marina campus, where you have all your sports facilities. We have a nice track, the athletic track. And I think you should visit all departments. And after that, probably some of the faculties will take you through MIRM. Most of you visited MIRM? No, right? Dr. Vinay or Dr. Shagufta will take you guys to the MIRM building and show the facilities in the second half of the day. So as we stand here, you should know a little bit of what Mahe is and what is its legacy and what we are as an organization. Only then you can sort of incorporate in our culture, or soak into our culture. So Mahe is the only private university who is in the NIRF ranking list. The first is 6th in the NIRF ranking, and all the 5 who are above the ranking are all government institutions like IISER, JNU, BHU. So all other private institutions are basically below in the NIRF ranking than Mahe. So you are actually sitting in a very privileged place. Apart from that, we truly believe in sustainability. And we, as Deputy Registrar has clearly pointed out, we are the first. We are the first in India to have the Green Matrix ranking. We have a very balanced gender aspect ratio, where around 60 to 40. So we believe in gender equality, not that we are against boys or girls, but so it's really balanced. So ours is one of those private university which boasts of this gender ratio, right? Both in terms of our students as well as our faculty requirement. You can clearly see that 50-50 dissection. So we have been consistently getting an A++ grade from the past 5 years, and we hold the institution of eminence tag. So definitely the institution of eminence tag comes with a lot of responsibility with respect to the quality of teaching we impart, the documentation and the precision we maintain with respect to conducting our academic activities as well as research activities. So all these tags give upon us a lot of responsibility as stakeholders, not only we as faculty, teachers, but also you as students, to uphold these values, right? And as we start our academic journey today, I give you confidence that we will take you through. Right now, all of you are looking a bit confused as to whether, whether we'll make it, how it is, but I'm sure by the end of this semester, all your doubts will be taken care of, and I assure the parents that we are there to mentor them, handhold them, and take them through the journey. So because of this, I think most of you would have got the mentor-mentee lists, and your mentors would have messaged you, right? So they are the people who will really be with you, and in case of any kind of problems, it could be academic queries, hostels, whatever, please get in touch with them, and then they will get you in touch with the relevant people. Okay. To tell a bit about what our programs are, we have the flagship program of our institute is the stem cell biology program. The stem cell biology program started in 2007. Okay, so the stem cell biology program, there are 2 stem cells programs. One is the master's in stem cell program and an advanced PG diploma program in stem cells. So the advanced PG diploma program is more for people who want to use this opportunity to know a little bit about stem cells. So the emphasis is more on practical training and a 3 months project work. Whereas the Masters in Stem Cell Biology program is a more comprehensive program spread over 2 years. The first year would have 2 semesters, and all our courses are modular in nature. What I mean by modular is like, suppose like what Shashidhara, Professor Shashidhara has told is like you start from the cellular level and then go to the organ level and then go to the organism level, right? Irrespective of whichever course you take, it is very, very important to develop this understanding about different concepts. So we will take you through that, to that understanding, irrespective of whatever course you take, by giving you knowledge in that particular modules. For example, there is a common module for all the process, that is the laboratory technologies. So technology is a very, very important part of learning now, it's a very, very important part of research. So the technologies are common, which you will be using for whatever kind of questions you are asking or whatever field you are choosing. So there's a common laboratory methodology course across all the programs, be it MSc in Stem Cell Biology, Translational Neuroscience, or Translational Immunology, right? And then you will have field-specific niche courses, which will take you through. So during the first half of this semester, you will finish up doing your basic courses, which is called as the Module 1 and Module 2. For example, in stem cell biology, you will have laboratory methodologies and biomolecules. Same is the case with neuroscience. In immunology, you will have fundamentals of immunology and laboratory methodologies. So the laboratory methodologies is a common course. Once the basics have been laid, then we go to module 3 and 4, which will be very subject specifics. But otherwise, the way the course is structured is we have our practicals integrated with our learning modules. For example, if you are doing your laboratory methodologies course and somebody is teaching you genome engineering, then restriction, digestion, cloning, those practicals will be integrated as a part of the practical curriculum for the laboratory. The same is the case with all other modules. So the MSc Stem Cell Biology course has a 1-year project work. It is more interdisciplinary in nature, where you have a tissue engineering component, developmental biology component, neuroscience component, and architecture, cell architectural components, and biotherapeutic component. So the stem cell biology course is a more interdisciplinary course. The translational neuroscience is focused on neurobiology and neurosciences. The translational immunology course integrates the basic and modern concepts of immunology for disease understanding. This is how the courses have been structured. Apart from that, the base program which we offer at our institute is the master program. As of now, we don't have any bachelor programs. And it's basically an academic research intensive institute, so our master's programs are also integrated with research. So we have a very, very strong doctoral program. All the faculty here are well qualified. They have done their PhD, they have done their postdoc in their niche areas, and they run their focused research groups in different areas related to the master's program which we have in our institute. So the students will be very much mentored, very, very intricately, very closely by our faculty and sub-mentored by our senior PhD students as they progress through their journey and as the course curriculum proceeds. So let me introduce you to the course coordinators for different programs. The course coordinators basically are involved in designing the timetable, designing the modules, taking care of the questions, organizing for the sessionals, and they played a very, very important part in sort of making the course together along with all other faculty. So let me introduce the course coordinators for MSc in Stem Cells, Dr. Shagufta and Dr. Meenal Francis. Can you please just stand up and, yeah. So for the advanced PG diploma in stem cells, again it is Dr. Shagufta and Dr. Meenal Francis. You will be interacting with them more closely, as in the next week, they will tell you like the syllabus, the schedule, they will share the schedules with you, they will share the practical modules with you. In a short while after I finish my presentation, Dr. Shagupta will be talking about how the exams are held, what are the times. We follow a very rigorous academic calendar. Everything happens on time, when the sessionals would be there, when the final exams would be there, when the practical exams would be there. All that is very clearly plugged in our academic calendar, along with the events which we keep having every now and then. Okay? So for the Masters by Research in Translational Neuroscience, it is Dr. Anand, Professor MIRM, and Dr. Urmi Bandopadhyay, who is a Ramalingaswamy Fellow, Ramalingaswamy Faculty Fellow. Both of them would be coordinating the course. They will be sending you updates about different things like the timetable, classes, examinations, and things like that. MSc by Research in Translational Immunology is co-coordinated by me and Dr. Uttara, who was the MC for the day. So I think you know both of us well by now. So for any kind of queries with respect to timetable, schedule, and things like that, assignments, these people would be the people who will be interfacing with you guys. What are the highlights of the MIRM courses? How is it different from the other master courses which are offered in different universities, right? Most of us, like I think your parents or people like us, even we faculty, had either biotechnology or biochemistry or microbiology, right? So most of them were focused courses, but definitely the MIRM courses have a edge over the other master's programs. It's not that I'm bragging about the programs which are there here, but we have a very good admixture of practical training, theoretical learning, and inquisitivity-based learning, right? I think that is what most of us who are older in the audience had missed that. And we took care that the course curriculum is developed in such a way that the students develop that sense of inquiry, they develop that interest. And it is more experiential in nature, more communicative in nature. You can see that most of your students would not be the ones who will come, flash their slides and go back. They will, in fact, put up things from their own research to enhance your understanding and learning about different concepts. And it would be very interactive, and it's now up to you as to how you would try to snatch that knowledge from our highly equipped and highly experienced faculty members. So we emphasize more on practical training, right? You can teach a lot about Drosophila and Drosophila genetics, right? But till you actually dissect out, till you see the strength of how Drosophila genetics can help you in understanding mechanisms, you can't appreciate the biology, right? So the way we impart learning is more experiential, where whatever is being taught in the theory modules will be shown in the practical modules. For example, if Dr. Shagupta or any other faculty is teaching you about induced pluripotency, there will be a practical module where you will get to culture iPSC cells. Similarly, if somebody is working on host pathogen, we are teaching host pathogen interaction, so how to culture neutrophils, how to set up a co-culture of a bacteria and a neutrophil, which Professor Shashidhara has talked about. So the emphasis is on experiential interactive learning. All the courses in this institutes are research integrated, right from the advanced PG diploma program where we have a project component, right? So the advanced PG diploma has a 3 months project component. The master's in stem cell biology has a 1 month project component, and the master's by research courses have 1.5 year project component. So this also gives you a choice to choose your field of interest. For example, in the first year, the basic learning and the basic foundation is already laid down for all students alike. And then suppose a student is very interested in understanding disease, like diabetes, right? What are the what are the mechanisms of insulin resistance. So there can be a faculty who can offer a module on chronic metabolic diseases, and you can actually opt for that module as an elective, and then 10 rigorous classes will be taken under that domain, and a project is conceptualized along it. So what is the advantage of this? With the new NEP policy, we are sure going to shift into a system where there will be 4 years of bachelor followed by PhD, right? So our master's programs are also structured such a way that first year would be theory and practical learning, but the second year is already into research. So if you like the problem, if you have nice leads, good leads, and if you are interested, actually you can pursue it as a PhD program later on, right? So you save on that time, which is difficult if you take other programs like master's programs in biotechnology, etc., where you have, till now, what I know about is they have 3 to 4 or 6 months of project work, okay? So there is flexibility of electives, and we have a very, very limited intake. I think till yesterday we were getting applicants, but we had to actually say no to it because we don't take everybody. We take people who are genuinely interested and who want to commit for our courses, because it's not necessarily the marks. It's also about how interested are you and how committed you are for learning and science. So that has been our emphasis while recruiting students also, or while interviewing the students. So we have a very good student-faculty ratio, like we are 55 of you, all of you who are here. Some of them could not make it today because they'll be traveling and coming on Monday. So there is one faculty who will take care of my students. So that's pretty much a very good faculty-student ratio. So that also says that we really value our students and we believe in individual mentoring. So we are there for them, for their, in fact, all our faculties, if I can say, the timings are 9 to 5, but we have students calling us even in off hours, asking for queries and things like that. So that is the kind of culture which MIRM has. So the other advantage of this kind of research integrated course curriculum is there is a provision where if you're good and if you are able to identify a problem, you can publish that part of the research. And for your SOPs abroad, where if you want to actually go for a PhD, they would ask for research experience. And if you are lucky and if you are able to manage a publication, it's going to be it's going to hold weightage when you put up your applications to universities across the globe. Apart from that, we also believe in practical training, so that is very advantageous because people in the industry are looking for trained personnel. They don't want to invest in take the person and train them on the job. They want somebody with a technically sound background, and that's where MIRM is going to have an edge because we give them real bench experience with respect to not only in the project work, even before that, they get to learn a lot of techniques and technologies which are very industry relevant. Our courses are very interdisciplinary. I think you would have heard Dr. Shashidhara telling the importance of interdisciplinary research. You can work from plant genetics to Drosophila model system, and then you can integrate those concepts to cancer mechanisms, right? So that is very, very important. If you look at carefully the course curriculum of all our courses, you can see that there is stem cell biology in immunology, there is stem cell biology in neuroscience, there is neuroscience in stem cell biology. So it is irrespective of whatever you are taking and whatever your basic interest is, you are integrating different concepts to understand a field. And all the faculty are committed to develop this course curriculum and impart this kind of knowledge. So we have a lot of initiative. This year is the year of research excellence for Mahe. So Mahe has come up with very innovative incentives for students. Now the students who are enrolled in the master program get an incentive for a conference fund. They are, if they want to go for a conference, they put up a abstract, and if it is selected, Mahe is going to fund them. So there's a fund of 20,000 rupees during the course duration, which can be used for registration, accommodation, and travel to these conferences. So this is something, and it can also be used for part payment of the APCs. So I request, and in fact, I implore all the students to be sincere in your classes, identify your problems for your project work right from the first year, and think through it. Then that will give you the time advantage in probably writing a review or this, then you can get these incentives. Okay? So for all students of all the programs, if they are able to publish a paper or a review article as a first author in the first year, there is a 25% concession in the fees for the next academic year. So all these things tell about the kind of commitment which Mahe has as an organization for encouraging research right from the undergraduate level, postgraduate level. So what are the MIRM strengths? We have very good experienced faculties. There is diversity in faculties, that is also very important. They come from all part of the world and all part of India. So with them, they get all the richness of their experiences, scientific, personal, cultural, and you can clearly see that Mahe is clearly a multiversity in that sense. Okay? So you should benefit from them. Since we also have the Bangalore advantage when it comes to our courses, because we are located in the Bangalore Biocluster, which constitutes of INSTEM, NIMHANS, NCBS, IISER, and most of our faculties are somewhere or the other related to these organizations. So with them comes the collaborations from those places. So the students are going to largely benefit with them because you can engage in collaborative projects. So the projects during your second semester and the third semester has to be necessarily be done in the institute, but the fourth semester project can be done in collaboration with other institutes too, in consensus with your PI. So the PI would have their own research problems where they will have their collaborators. So a part of the objective can also be done in these institutions. So we have large number of international and national collaborations, MOUs have been signed. So the students largely get to benefit with respect to training and exposure from all our partner organizations. So we really, as per we follow the NEP policy, we believe in outcome-based education. So everything which you learn should have value in terms of research or in terms of deliverables, in terms of translation. So we incorporate all that as a part of our course curriculum. So the research specializations are very diverse, right? But all of them are asking questions related to stem cells and regenerative biology. So regeneration is a key thing. Regeneration, injury, repair, right? Would not just require an immunologist or a neurobiologist or a developmental biologist. You require expertise of all these people to regenerate tissues, right? So we have this admixture of expertise available in our institution. We have developmental biologists, we have tissue engineers, Dr. Shagufta, Dr. Anujit, they work on developmental biology principles. Dr. Manasa is a tissue engineer, Dr. Anand, Dr. Anujit, Dr. Sangeeta, they ask neurobiology questions. Myself and Uttara, the research focuses on immunology and regeneration and repair mechanisms. Dr. Uttara works on host pathogen interactions. Dr. Urmi works on autophagy and cell biology questions, talking about organelle quality control mechanisms. So is Dr. Sangeeta. Dr. Vinay, he's into epigenetics. He's trying to understand the epigenetic mechanisms of myogenic differentiation. Most of us work on metabolism and aging-related disease, all of us, all the faculty here. Dr. Meenal works on pancreatic biology and diabetes. I hope I have not forgotten everybody, right? I have included all the peoples, right? All the areas which are there in our institute, but you will get to know more about them when they teach in the classes. So the classes which are allocated to each faculty is based upon their expertise. So when they teach in the class, they just don't teach theory. They also bring to the table or bring to the desk their richness of experience from their publications, from their research focus, and they give anecdotes of those when they are imparting the training. So that is one USP of the courses in MIRM. So we are hugely funded, as I told you, like that the base program is the master program. The base program is the master program, and then we are a research institute with a very, very strong doctoral program, and doctoral programs and research programs have to be supported by funding, right? Otherwise, they'll be lost. So we are very, very hugely funded, and all our faculty, all our PIs have funds to run their research labs, and definitely the master students are going to benefit from that because the project funding comes from these grants. So we don't work in isolation. We have a good combination of basic biology, fundamental biology, applicative biology, and industry-relevant R&D too. There might be some of you who would want to get into applicative biology or go into R&D sector. So with that, we have industrial collaborations. Recently, very recently, we have signed a MOU with NCure Therapeutics. It is a NK cell-based oncotherapeutic company, and we are going to engage in research, and it's going to definitely benefit people from all fields who would want to ask questions related to regeneration or immunology. Apart from that, we have our tie-up with SciTech. So they have kept their imaging flow cytometry platform in our institute free of cost, so that we have an agreement with them. So the students get to understand these cutting edge skill sets. They get to do the hands-on on these. So these are things which are not available in all institutes, right? So please make use of the facilities which are there in MIRM. Get wet, go get your hands wet, explore. I think like even after the practical hours, if you go to our faculty and tell, I want to learn it more, I want to actually understand this technique more, I don't think any faculty will say no. And that is the culture which we have at MIRM. We have a lot of clinical collaborations because ours is a very translational institute, or our courses are very translational. So we collaborate a lot with the clinical community because that is where we get our biological samples. And it's a nice interaction between what is the unmet need in the clinic and like what the clinicians want in real life and what are the problems around which our researchers can work. So many of our faculty have strong clinical collaborations and they run projects with clinical collaborators. So the master students are going to definitely benefit with these kind of associations. Apart from that, we have Mahe as such has a lot of international collaborations. A broad MOU has been signed with many universities, Australian universities, UK universities, Australian universities, and we have sub-agreements with these universities to run focused research programs. Some of our faculty, they are running their research programs with the University of New South Wales and the ICAN School of Medicine. So the PhD students, co-supervision is allowed here. So some of our faculties are co-supervising their PhD student along with a collaborator from some of our partner universities. So there are a lot of student engagement activities. It's a nice admixture of fun, entertainment, engagement, knowledge, upskilling in the institute. So we have, from time to time, we have workshops because upskilling is a part of learning, because what you would have learned when we were actually doing our masters, we used to run an agarose gel, right? But now we can do a PCR, we can look at amplification in real time. The real-time systems were not even there, right? So definitely from time to time, upskilling is very, very important if you want to change with the world. Right now AI has come, AI is into healthcare. You can't say that I would not learn AI, right? Because you need to statistical tools. So from time to time, we have we have these upskilling workshops to make sure that you catch up with technologies, and generally they happen every half yearly for sure, sometimes even quarterly. Recently we had 2 workshops. One was on imaging flow cytometry, which was run along with our partner universities in Australia, and Dr. Paul Wallace and Dr. Uttara had run the imaging flow cytometry workshop. The tissue engineering workshop was run by Dr. Manasa along with SciTech along with AVE. Along with AVE Biosciences. So even the other faculties are going to have these programs from time to time depending upon the kind of expertise, right? So this is going to help you when you're going to apply for jobs because you're already trained. We are going to start with new certification courses in imaging and microscopy. Mahe has been really generous to give us funding for a confocal imaging microscope. So we are going to develop a bioimaging facility. So we have the state-of-the-art infrastructure in Mahe MIRM. There's nothing which you don't have at a master level, right? So it's up to you as to how you will utilize these resources for learning and for your project work. So apart from that, we have all these cultural activities. Routinely, we have talks and webinars from experts like Professor Shashidhara who talk on specific topics, and like, you know, the students are free, master students, PhD students, researchers, everywhere. Everybody can actually engage in interesting scientific conversations. And if they like your question, they might just come and say that, why don't you join my lab, or why don't you apply for a PhD in my institution? So we create that we create that ambience in MIRM to foster these kind of collaborations and recruitments. So as a part of our initiative to engage both globally and nationally in research and to exchange our ideas, the research which we do, we are going to have an international conference in August 27 to 29th. We have almost the entire India, the best scientists in India and abroad coming and speaking there. I implore all of you, I think most of you would have got the information regarding the international conference. I think you should register for it. We are a small institute, not necessarily small with respect to our expertise and strength, but in terms of numbers. So definitely we need people, volunteers who would help us in this in organizing this huge conference. So I would want some of the youngsters to actually come up, register, and also help us in organizing this conference. So it's really something which is very inclusive, and there are no boundaries, and people talk to each other and work together, right? So on this note, I would, on the behalf of faculty, students, and staff of MIRM, I would wish the new batch good luck. Thank you. If you have any questions, quick questions, after that, Dr. Shagufta will be talking more in the micro details about the different courses, the modules, the examination pattern, etc. Probably we can take questions then, right? Yeah. Thank you.
Thank you, Dr. Jyoti, for the elaborate description of the course and introduction to our faculty members. I want to call upon Dr. Anujit, our Professor MIRM, to hand over a token of appreciation to Dr. Jyoti. Request Dr. Jyoti to come on stage. Now I want to call upon Dr. Shagufta Parveen, Course Coordinator MIRM, to give us a description of our syllabus.
Hello, everyone. Dr. Jyoti has already given an overview regarding which are the courses that we have in MIRM. I will only give details regarding the subjects which are involved in each of these courses and the examination patterns. So, as you know, we have 4 programs at MIRM. MSc in Stem Cell Technology and Regenerative Biology. We have an advanced postgraduate diploma course in Stem Cell Technology and Regenerative Biology, MSc in Translational Neurosciences, and an MSc in Translational Immunology. So the first program, this was the first program that was started in MIRM, MSc in Stem Cell Technology and Regenerative Biology. It is a 2-year program. It is divided into 4 semesters. The first year is theory and practical teaching. The first semester has 4 subjects, or modules as we call them. Biomolecules, laboratory methodologies, architecture of cells, tissues, and organs, embryonic differentiation, embryonic development, and cell differentiation. There are associated labs with these 4 subjects. As you can see on this slide, a few credits are associated with each of these modules or subjects, and the final marks for each of these subjects is out of 100, of which 30% of the marks are internal assessment and 70% of the marks are a final exam or an end semester exam. For the practicals, the distribution is 40 and 60, and it equates to 100. We have divided the classes in 2 blocks. The first 2 subjects will be taught over a period, over the first 2 months, and the second 2 subjects will be taught over a period of later months. So in August and September, we will finish the first 2 subjects. October to November, we will finish the second 2 subjects. Normally, all your classes start at 9:30. We have 2 classes in the morning, 2 theories, each for 1.5 hours, and the afternoon session is practicals. The second semester also has 4 theory and 4 practical subjects. We have intercellular communications in stem cells, biomaterials and tissue engineering, clinical application of stem cells, and non-clinical application of stem cells. Each of these has an associated lab paper also with it. Again, there are a set of credits and how the marks are divided. The classes are again, as you can see on this slide. The second year is project. It is divided into 2 semesters. The third semester will have a short theory component, a practical component, and a mini project. Again, each of these have associated credits with them. The fourth semester is only the project, and the marks are specified on this slide. We have the Advanced PG Diploma Program, which is a 1-year program. It is not divided into semesters. It is a continuous academic, 1-year academic program. There are 3 theory subjects, 3 practical subjects, and a small mini project, which is a part of this program. The mini project has to be defended in a viva voce exam. The third program that we have is Translational Neurosciences. It is an MSc by research, again a 2-year program. The first semester is theory and practical teaching. In the MSc Neuroscience program, the first 2 subjects are shared with the MSc Stem Cell Technology and Regenerative Biology program. So the students will be doing these classes along with the MSc Stem Cell Technology program. The second 2 subjects, Fundamentals of Neurosciences and Central Nervous System Development and Disease, is exclusive to this translational neurosciences program, and it will be these subjects will be taught separately. There are 4 associated labs with it. The second semester starts with the project. There is a theory, small theory, practical component to it with a project. The third semester is also similar, and the fourth semester is exclusively project work and dissertation. The MSc Immunology program is again by MSc by research. It has, the first semester is theory, practical teaching. There are 4 subjects in this, subjects or molecules. Fundamentals of immunology. The laboratory methodologies is shared by all the MSc programs. Principles of regeneration, immune dysfunction, and disorders. There are associated labs with it. Second semester starts with the project. Third semester is the project, and the fourth semester is project exclusively. On those slides, you could see that there is a internal assessment and a final end semester exam marks which you get. So the internal assessment, we do daily assessment, so you have some marks which you get there. We have sessional tests or middle semester tests. So those marks are combined and we make them into 30%. That is 30% of all the total marks that you get. Practicals is 40%. The minimum marks for pass in the university exam is 50. You must score 50% separately in the internal assessment and in the end semester exam. We want you all to attend all the classes without missing, but in case you have some kind of a medical problem or it is inevitable that you have to miss classes, we allow you to sit for the final exam if you have a 75% attendance. Anything below that will not be entertained. We expect you to clear all the subjects in each semester within that semester itself. However, if it is just not possible, you can go to the second year by carrying over some subjects from the first year. You will not be allowed to defend your viva voce dissertation mini project unless you have cleared all the theory exams and all the practical exams of the 2 years. As Dr. Jyoti mentioned, some students will be allotted to faculty, so there will be mentors and mentees association that will be going on, and if you have any problems, you can contact your mentors. There will be a student council to address academic issues. This will comprise of the head of the institute, a chairperson, and 2 student representatives, a boy and a girl, and this committee will update the faculty regarding any problems related to academic issues before the exams. There are student counselors, and there is the Office of Student Affairs, which is there in the Mahe campus if you have any other problems that have to be dealt with. Again, I welcome all of you to MIRM. Thank you.
Thank you, Dr. Shagufta, for the elaborate syllabus description. I want to call upon Dr. Anand. Is he around? No, looks like he's not around. I'll call upon Dr. Jyoti to hand over a token of appreciation to Dr. Shagufta. Now I would like to call upon Dr. Uttara Chakraborty, our cultural coordinator, to give us a description of cultural activities and other activities that go on at MIRM.
Thanks, Meena. I'll keep it very, very short and kind of a relief to all of you. It's not that we are a very, very heavily, course-filled curriculum that you have landed up in, and there is no kind of event or extracurricular or co-curricular activities that you would be getting chance to work with. It's not like that. So I just thought this is the occasion when I will briefly introduce to you that MIRM is also socially, culturally very, very active and conscious. So to begin with, we'll talk about the rewilding team or the rewilding task force, which is essentially built in the Mahe Bengaluru campus, spearheaded by some extremely vibrant scientists from different departments, especially from Srishti School of Art and Design. And MIRM is very happy to be a part of this rewilding task force. So what does this do basically? If you've seen this campus, this is a huge campus, and a campus which is very much rich in flora and fauna. So it's one of our tasks to also preserve this flora and fauna in this campus. And as a part of that, there are various activities which this rewilding team does. For example, like bird watching or bird counting. And in night activities for frog observations here. There are various species of frogs which are actually available in this campus. The entire motive of this rewilding task force is not just restrained to the flora fauna, but basically we have taken up a task, which is a huge task to rewild and transplant trees in this campus. There are regular plantation drives which have started and which we are planning to engage you students into in regular intervals, where you will get opportunities to plant trees and have your own small land which you can call as yours, which you can nurture and develop. So this rewilding activity is something which definitely MIRM would introduce all of you to you gradually, and you will be an integral part of this activity here. The other activities, to name them, this newsletter which in the morning you saw was launched, is something which we encourage all our students to contribute to. Science and research is integrally a field where you constantly need to write and document. And if you have or inculcate this culture of writing scientific articles, then the newsletter or the Rejuven is actually one of the best platforms that we give to our students to express yourselves. Any scientific feat, be it in your field, be it in others, you can actually talk about that. And as you saw, that many of our students have come up with such articles and ideas of their own. The second, as Meenal mentioned, so we have a cultural committee in MIRM, and the Rubaru is involved in organizing the annual cultural event, along with the regular regional events that are constantly going on. And the cultural event definitely would want participation from each one of you to come up and show your potential, cultural potential, which you can actually showcase, make use of, and make us all feel proud of. So that is something which we would definitely want you to engage with, apart from studying and apart from all the courses that you are going to be getting into. And we also have journal clubs, which is an essential part of a research institution. Our PhD students are engaged in presenting their work or any scientific stream that they would like to talk about, and we encourage all our students to participate in such programs so that you can get ideas, you can ask the right questions by looking at research from various and different other fields and different labs in MIRM. So this scope is something which you should definitely, work on and grab, as one would say, because this is something which you essentially wouldn't get all under the same roof. So you get your degree, your master's degrees, but parallelly, you get so much inculcated into research and scientific research of various backgrounds that really goes a long way with all our students. The alumni have all given us excellent feedback of the kind of training that they carry forward with them when they go or move on to the next institutions where they want to build up their careers. So with this, I think we can I would like to stop here and move on to the last session of today's program. If you have any questions, we can definitely interact afterwards to clarify any doubts from you. Thank you.
Thank you, Dr. Uttara, for the colorful description of our cultural activities. I want to call upon Dr. Jyoti Prasanna to hand over a token of appreciation. Please come on stage.
Actually move on, we also have a sports day in our institute. So we have our sports coordinator, Dr. Ramanjot. Can you just stand up? We have our SLCM coordinator, Dr. Anujit, takes care of the digital entries and things like that. We have our web coordinator, Dr. Vinay. And then we have our social media and social engagement coordinator, Dr. Shweta. Dr. Shweta is not around. Yeah, okay, thank you.
Now I want to call upon Dr. Arjuna, our Assistant Director, Student Affairs, to come and give us a description of student support.
Okay. Good afternoon. Thank you, ma'am. Yeah, I am Dr. Arjuna. I am a faculty member at Department of Liberal Arts, Humanities and Social Sciences at Mahe, Bengaluru. And also I'm taking care of Department of Student Affairs at in this particular campus. So we are total 7 members team with 4 student counselors. Oh, okay. Okay. These technical glitches are not in our control, so we are used to it because of the COVID and other things. Okay, so we are 7 members team with 4 student counselors, Miss Lakshmi, Miss Navyashri, Miss Shailaja, and Mr. Aditya. And we have 1 student experience consultant, Miss Saujanya, and our office admin, Miss Nayana. We all are here to support you. In what sense? Why we are here? To support on these 3 aspects. First, student engagement. Second, extra and co-curricular initiatives, which rightly pointed out by Dr. Uttara ma'am. And mental well-being support, which is very, very important. So there will be many kinds of concerns which we all go through. College and mental health concerns are one of these. There are, I mean, this particular department, MIRM, is research oriented mainly. So in that process, you might get into stress. you might have to manage time, and these are the common things. Anxiety and these kind of things are not in our control. And in those days, in earlier days, it was not addressed carefully, but now we all are aware about all these concerns, which are kind of obstacles for our goal, reaching our goals. So we will be supporting you in handling these kind of concerns. Psychological well-being concerns are also addressed by our team. Behavior concerns also. It's not like you have you have to meet student counselors or student experience consultant if you have any problem. If you have a unique idea which can be implemented at the university level, you are welcome to meet us. And if you have some idea to improvise your personal behaviors, or if you are struggling with some minor problem, which is not necessarily it has to be a mental health thing, but behavioral related things also can be supported by our team. So these kind of mental health expertise are there with our team members. The DSA plans for 2024 and 25 is this. There are many workshops, seminars, and awareness sessions are planned related to managing exam stress, addiction awareness related thing, gatekeeper to prevent suicide, these gatekeeper sessions to prevent the suicide attempt or suicide thought and suicidal thoughts and other things for faculty, staff, and also for peers. We have self-care fair planned for the World Mental Health Day. We have psychological and medical first aid things. These are some of the mental health related things. But we also have gender champions for gender inclusivity. We have student club initiatives, which in all the respective departments we have student clubs. We will be closely supporting them to do some cultural and extra or co-curricular activities. We have higher education competitive exam counseling sessions also will be arranged. And Mahe Bengaluru reads, Mahe Bengaluru paints, knits. If you are passionate about all these kind of other very creative activities, we will be providing a platform where irrespective of department, irrespective of your programs, you can come there and do all these kind of activities. We will be having weekly open jam. If you want to no need to be Arijit Singh or Sonu Nigam or Shreya Ghoshal. You can just come there and sing if you want, if you are a singer. If you want to just perform in front of the interested audience, there won't be anyone to judge you. If you are a instrument player, you can come there and showcase your talent. So that particular platform will be there exclusively for students. Of course, faculty can also join us. And also, interestingly, who doesn't love watching movies? All of us, right? So we will be watching some of the interesting and very nice movies just to relax. If you want to discuss upon that particular thing, not in a very technical way, but in a very fun way, we will be doing those things also. So even if you are a social person, if you want to contribute to the society, we will be having some events with the nearby villages and other places where you can be part of us and be part of those kind of initiatives as well. So these are the very, very fun and creative, of course, very important, like mental health related concerns, which we take care of. And DSA also looks into this anti-ragging thing, which is very, very important. In India, and of course in Mahe, in all our campuses, we have a zero tolerance policy on ragging. So don't use your energy into these kind of things. Don't ever get into such things. It is very, very problematic. I'm clearly telling you. You all are aware about this. You are at the PG level. You know about this in a better way. But don't ever think about all these things. We are very, very strict on these kind of things. So in the Anti-Ragging Act, which is presented by UGC in 2009, there is, in the third amendment, recent amendment, it was clearly mentioned that any act of physical or mental abuse, including bullying and exclusion, targeted at another student, fresher or otherwise, on the ground of color, race, religion, etc., is considered as ragging. There are a few cases where addressing someone with inappropriate way, hey, XYZ, or in this way also is considered as ragging. Believe it or not, it is true. Because if the other person is not convenient, feeling good with that kind of expressions, if he or she is expressing that, there is an opportunity to tell that particular person, don't do that. I'm not liking it. I'm not okay with that. Please don't do that. If that other person is still repeating that, you can report those things as ragging. And it's not necessarily you have to come to us, okay? You have, each and every respective departments in Mahe, Bengaluru has anti-ragging committee, which is headed by respective teachers. And HODs and HOIs are also very well aware about the importance of this zero tolerance on this particular aspect. So don't think about it. Of course, you don't do that, but still, don't use your energy on these kind of things. So punishable acts of ragging, there is a list given by the UGC in their official platform. So any kind of thing, it's not necessarily, it has to be a physical thing. Physical or psychological humiliation is also one of the punishable acts of ragging. So what are the punishments? Depending upon the nature and gravity of the offense as established by the anti-ragging committee of the institution, the possible punishments are listed down in that. We will take all these, as I mentioned, ragging is an important matter. If there is an incident, Mahe never took it in a lighter note, okay? So these are, there will be fine, there will be collective punishment and so on and so forth. So don't engage yourself in this kind of act. Whom to contact? Respective teacher, respective, in all the departments, there is one student affairs coordinator. In MIRM also, Dr. Sangeeta Nath is the coordinator as of now. So you can reach out to her. Even Dr. Jyoti Prasanna is also, will be ready to listen to your concerns. If not, you can come to me or any of my teammates, or you can directly approach Deputy Registrar of this particular campus, or even Pro Vice Chancellor. If you want more information about this, you can log into antiragging.in, which is a Ministry of Education, but UGC initiated thing, which contains complete information about it, okay? Another important aspect is grievance redressal. Mahe Bengaluru has a robust grievance redressal committee to cater to student grievances and allegations of unfair practices, if any. The whole system at this particular campus is very well set. There won't be any chances for that. You can see when you have to enter the premises, you have to wear a tag. I mean, the ID card is mandatory. So many systems we have put in for the safety and security of our members of this particular campus. But if there are any unfair practices, if you, not necessarily it has to be experienced. If you are witnessing such things also, you are free to express that to your respective department or to us. So in all the departments, as I mentioned, grievance redressal committee is also there to address any kind of students' grievances, okay? So the members of the committee are nominated by the chairperson of the committee, depending on the type of grievance submitted. This is chaired by me as an assistant director, and the anti-ragging committee is headed by Pro Vice Chancellor. It involves the officials from the government service also, like police and other people are also involved in that. So in the grievance redressal, all concerned people will be involved in that particular committee. So what are the steps? What is the mechanism? How it works? So you can submit your grievances online or offline through email, WhatsApp, or personally you can meet us and you can inform that. Complainant called by DSA, application will be forwarded to Mahe GRC with a copy of Ombudsman. Ombudsman is the highest authority who will oversee the complete process. In this particular campus, Deputy Registrar is the Ombudsman. And Mahe GRC conducts inquiry, then communicates the decision to complainant through email and also to the HOI of the institution. If complainant is dissatisfied with the GRC's decision, he or she can submit an application to the Ombudsman. So then he will take some time to go through that particular thing and make sure this particular grievance is addressed, that is solved. Okay, so this is the important thing. And another important committee is Internal Complaints Committee. It is there to prevent or discourage any kind of sexual harassment in this particular premises. On this matter also, we have zero tolerance. I am not saying for the sake of saying it in front of all of you. We have taken care of all these kind of things in very, very careful manner. We haven't spared any of the culprit. We have expelled whoever is involved in ragging or in sexual harassment kind of things. Okay, so we are not informing you for the sake of thing. We are practicing it. There is an internal complaints committee, and it focuses on prevention of sexual harassment at workplace for women and girls mainly. And you can directly write to icc.mahebl@manipal.edu if you have experienced or if you have witnessed something. Okay, so don't hesitate. You are free to raise this particular issue to others. And also for the students, we have library facility in our campus. You might be aware about that by now. We have Marina, which is an indoor sports complex along with the gym. We have sports facility. We have, as already mentioned, there are sports coordinators in that. We have strong cultural activities are also involved in that at the university level, and also we support our students to go and participate in inter-institutional, the other campuses, you can go to other campuses to be part of those things. And we have CSO, Chief Student Officer, who look after the hostel and other services as well. And we have general services, which is taking care of transport, security, and other important services in the campus. All the officials of these services are always available for students. You can reach out to them through mail or through mobile, because the numbers will be shared from the respective institution. All the important contact details will be shared with you. Please check, students particularly, please check your learner ID, the university email, where you will be receiving all the communication related to Department of Student Affairs or Chief Student Officer's office or general services thing or library or all these services are there exclusively for students. So make use of all these things, okay? And finally, we are here for you, okay? These are the contact details of our student counselors and student experience consultant. And if we are all occupied in one or the other work, our office admin will be there. You can drop any information to them as early as possible. We will be happy to contact you. And in case of emergency, please kindly contact Mr. Patrick Vass, who is Chief Security Officer. And this number is not just, it's not his personal number. It is an official number, which will be active 24/7. And we have security cameras around the campuses where we might also not aware, but everything is closely monitored, not to check, keep an eye on you, are you doing your work or not, just to make sure you are all safe, okay? So anything, if you come across any emergency situation, you are free to contact this particular number, or you can write in if there is an incident happened, you can update him through email as well. So, and very soon, in a couple of days, we will be moving to the DSA team, particularly the student counselors will be moving to SSC, Student Support Center, which is little away from academic block to make it very comfortable for students. It's a very wonderful place. You will definitely like it. You can come there and relax. You can discuss, you can meet our student counselors, and you can interact with them. You can, if you have any good idea which can be implemented at the university level, some clubs at the central level which is useful for each and every students, those kind of things will be encouraged. Ms. Saujanya will be taking care of such things. So you are free to do that along with your academic duties, of course. That comes at the primary. Yes, once again, we welcome you all to Mahe, Bengaluru, and this is my contact detail. You are always free to reach out to us, and you can follow us on social media as well. And thank you so much to Dr. Jyoti and her team for making me and our team to be part of this particular wonderful orientation program. Thank you, and if you have any questions, you can ask. Thank you. You can take a screenshot of this if you need. That is also an important one. We will be sharing a separate email to you with all the details along with this particular slide as well. Was it a long day? But most of them were academic related. These are non-academic related things. So, and very, very important. It is exclusively for you students. And many parents also are here, I can see. So this kind of information is very important for them as well. So, no questions? Okay, thank you so much. Have a nice day.
Thank you, Dr. Arjuna, for giving us a view to all our students' support that our new students can avail. I want to call upon Dr. Jyoti Prasanna to hand over a token of appreciation to him. Dr. Arjuna, please come on stage. Many of you may be awaiting the end of the program or what is happening next. Now, it's lunchtime and it is the end of the program. I'm here to give the vote of thanks. So all things must come to an end. But this is a special occasion that ends with a new ARAMBH. I'd like to take this opportunity to welcome our new students to the world of Manipal Academy of Higher Education. Along with Mahe family, our Pro-VC, our Deputy Registrar, our Chief Guest, the Dean and faculty of MIRM, along with our research scholars and students, I want to extend a special welcome to our new students. I'd like to thank our Chief Guest, Professor Shashidhara, the Director, TIFR-NCBS, for joining us in welcoming our students and for a very inspiring talk. A special thanks to our Pro-VC and Deputy Registrar for always supporting MIRM in all its activities and the encouraging words from our Registrar for this new beginning. A big thank you to our Dean, Dr. Jyoti Prasanna, for all the efforts she put in from the time of admission to accommodating all aspects of our new aspirants and make this occasion, the new ARAMBH, a grand success. Thank you to Dr. Arjuna from Student Affairs, for all the supporting information our students require for this day and henceforth. Thank you, Dr. Uttara Chakraborty and Dr. Shagufta Parveen, for organization and introduction to the syllabus, and all our faculty members and research scholars for your presence in welcoming our new students. A special thanks to our research scholars Akshaya, Shritama, Jeevan and Purvi for their tireless efforts to gather the schedule together with a very well-designed poster and layout, and your commitment is greatly appreciated. Our thanks to our administrative head, Mr. Harish, for organizing and making all arrangements for this day. System admin, Nandish, ground coordinators, Basappa and Marappa, for all their support. At last, but most important, a big thank you to the parents of our new students for bestowing your faith in your children's choice of joining Manipal Institute of Regenerative Medicine. Once again, wishing all our new students a grand success and a sparkling future ahead. Thank you, one and all. All of you, I welcome you for a lunch get-together. Our ushers will coordinate with your places for lunch. Thank you.